series. So I am Dr. Akriti Tyagi. I'll start the discussion of the zoology paper first and so preceded by the other faculties for the respective subjects. So what we are going to take up today is the AIRTS minor test 2 discussion first. So let me tell you in brief what we are going to take up in this test series. We have already covered the first minor test. So this is already done and I hope you all have seen your result. You all have gone through this test and you know how NEET paper comes. That is in sync, is in resemblance to that paper that you are going to take up. You are a NEET aspirant, so have to go through it. The next paper, what we are going to take up now is minor test 2. And for zoology, the test includes the chapters like excretory system, neural control and coordination, chemical control and integration. So these are the chapters which comes in this. And so it is preceded by other test papers and then minor test 3 and 4 followed by some mega test that will include partial papers. They are not complete papers, complete chapters or complete 11th class syllabus in them, followed by the full test paper which includes the complete syllabus tests. So these are the dates of the respective papers. Try to be in sync with this as much as you want to be in sync with your NEET preparation. So let's just start with going through the questions of the very first session. So the first session is where all the questions are compulsory. There is no optional question in it. All the 35 questions, 30 questions are to be answered by all of you. So let's start with the very first question. The first question says, which of the following toxic waste can be removed with the loss of minimum amount of water? So the keyword here is loss of minimum amount of water. And your options given are ammonia, urea, uric acid, creatinine. And you know the answer would be option number C, uric acid. Why? Because it requires less and least amount of water. The maximum amount of water is required by ammonia, followed by urea, and then uric acid requires least amount of water. And this is how you have to remember this in lineage, right? So the answer is option number C, which is uric acid. Let's come back to the next question, question number 2, and the question is, among the following animals, identify the one that is not ammonotelic. This is highlighted because this is a word, it is not ammonotelic. And the options given are cartilaginous fishes, bony fishes, aquatic amphibians, and aquatic insects. And it is given in NCERT. Be in sync with NCERT, right? It is given there that bony fishes, aquatic amphibians, and aquatic insects, they all are ammonotelic. Right, they all are ammonotelic. And the question is asking for the animal which is not ammonotelic, and the answer is cartilaginous fishes, which are ureotelic. So the correct answer is option number A, cartilaginous fishes. The other way around is, if you need to remember, there is a shortcut to remember this. If you don't know the animal, then please go by it. The examiner always gives this word aquatic to make you understand that somehow the aquatic animals that lives in aquatic environment, habitat, they have ammonotelic nature of excretion, right? So you can exclude these two options based on this information. That is just for your understanding. Now let's go back to the next question. The question number 3 stated that column of Bertini, this is the key word, is an extension of. And the question is from the chapter excretory products and their elimination, and it is based on the diagram which is there in the NCERT. Column of Bertini, which is written here, is renal columns. This is column of Bertini, also known as column of Bertini. So these are actually the columns which are present or extensions of renal pyramids, these. So this is your renal pyramid, and in between them there are renal columns, right? So the options here are medullary pyramids, which are there, hilum, it is a notch here, this is your hilum at which all these things are present, the pelvis and the renal cortex. So this is the renal cortex. As we know, kidney is divisible into cortex and medulla. So this is your cortex, and inside you have your medulla. So this is the cortex that extends in between these medullary pyramids to form column of Botany. So the answer would be renal cortex. Renal cortex are actually creating the extensions in between the medullary pyramids to form column of Botany. Please remember it. It's a very important question. The kind of questions has been asked in previous year questions in NEET. So pay attention to this information. Let's move on to the next question that says that the Malpighian body. Now here please remember it is written Malpighian body, not Malpighian tubules. That's a different word, right? So you have two words. One is Malpighian tubules and one is Malpighian body. Both are excretory structures but in different animals. You will find excretory tubules in the animals called arthropods or insects, and you will find this word Malpighian body in case of man or human, right? So both are excretory structures but present in different animals. And here the question is asking for Malpighian body. Malpighian body, that means we are talking about human kidney. It does not compromise of which of the following. So Malpighian body is what? We know the unit of this human kidney, that is nephron, comprises of the glomerulus and the renal tubule. That glomerulus comprises of this, and this is your Malpighian body. This Malpighian body comprises of your Malpighian capsule or this Bowman capsule comprising of the glomerulus, that is the capillary which is formed by the extension of this efferent arteriole that enters into this concavity and then comes out as efferent arteriole. So this glomerulus, the capillary network, this is the capillary network. It, along with this concavity that harbors it and the Bowman's capsule, makes the Malpighian body, also known as renal corpuscle or Malpighian corpuscle. So what it all includes and what it doesn't include, let's go to the option first. The very first is inferior vena cava, second is glomerulus, as it includes this cannot be a correct answer. It comprises of Bowman's capsule, so this cannot be a correct answer. As it comprises of concavity of the Bowman's capsule, so this cannot be a correct answer because all these are there in forming the Malpighian body. So the correct answer is your inferior vena cava, because it is the arteriole, efferent arteriole, that enters and forms the capillary network in the glomerulus in the Bowman capsule and not inferior vena cava. So the answer is option number A. I hope this is clear to you. Let's go back to, let's move forward to the next question, which is question number 5, that states which of the following is not considered as a part of nephron. And the options given are distal convoluted tubule, collecting duct, proximal convoluted tubule and Malpighian body. So the nephron, which is the unit of this system, our excretory system, it comprises of your Malpighian body and then rest of the part. That includes your Bowman capsule, that includes your PCT, that includes your DCT and your loop of Henle. Right, so all these are included in the nephron. Not, what is not included is your collecting duct. Collecting duct is not a part of nephron. Please remember it. It's a very, very critical question. It's a very important question that collecting duct is not a part of nephron. Please write it down. Note it down. Collecting duct is not a part of nephron. Very important, right? Whereas distal convoluted tubule, proximal convoluted tubule, Malpighian body, they are the parts of nephron, so they cannot be the correct answer. And the correct answer to our question is option number B, collecting duct. Let's move on to the next question, question number 6 of this paper, that states that you have to read the following statements and identify the incorrect statement. So first of all, whenever you see such a long question, as it's a difficult question, right? It is a difficult question. It's not an easy question to do. So whenever you do such kind of questions that includes multiple statements, first of all is always mark what is being asked in the question, as the question is asking for incorrect statement, because when you go through all the statements, at the end of it you will not remember whether you have to choose the correct or incorrect statement. So always go back to the question before answering it. That is for the long questions, for the difficult questions, right? So although it is a difficult because it is lengthy, but conceptually it is very easy and directly from your NCERT lines. And the question is about the volume of blood that is filtered by the kidneys. So it says that the volume of blood filtered by the kidneys per minute is approximately 1/3 of the blood pumped out by each ventricle of the heart per minute, which is incorrect. And we know these diseases. Now please understand, this question can be reframed in multiple kind of questions based on the information given in it. It is very, very important. The keywords where you have to pay attention to is whether it is written it is filtered by kidneys or kidney. This S is important, whether it is one kidney that is filtering it or two kidneys. Whether it is per minute, per second, per hour given. Units are important. Second thing, it is 1/3, 1/5, 1/7, 2/3. This digit is important. Third thing, it is pumped out by each ventricle or both the ventricles. If this is written, this is also incorrect. Next keyword is heartbeat per minute per second, and again the unit is important. And based on this is incorrect because it is 1/5 and not 1/3 of the blood that is pumped out, because we can relate it to the cardiac output, which is making it as 1/5 and not 1/3. The second statement is the volume of blood filtered by the kidneys per minute, it is correct, is approximately 1/5, this is correct, of the blood pumped out by each ventricle, this is correct, of the heart per minute, this is also correct. So this would be the correct statement for this. But as we have to find out incorrect one, let's see what it is. So you basically have to find the correct statement here. Second is the volume of blood filtered by kidneys per minute is approximately 1/5 of the blood pumped out by each atrium. This is also incorrect. And the last is the volume of blood filtered by kidneys per minute is approximately 1/3. This is also incorrect. So the correct statement here is option number 2. So B is the correct answer for this question. Let's move on to the next question, which is question number 7, that states glomerular filtration rate or GFR in a healthy human is approximately. That's a direct question. It's an easy question. And we know the answer is 125 ml per minute or 180 liters per day. So this is the digit you have to remember, and based on this the answer is C. So as I'm stating, units are really, really important, right? So pay attention to whether it is written per hour, per second, per minute, per day, meter, liter, milliliter. These units are critical. So the very first option given is 180 liter per minute, which is incorrect because it is 180 liter per day. 125 ml per day is incorrect. It is 125 ml per minute. This would be the correct answer. This is incorrect because it is per minute and not per hour. So the answer is option number C. Let's move on to the next question, which is question number 8, that states approximately -% of renal filtrate has to be filtered by the renal tubules, and we know it is 99% of the filtrate gets reabsorbed by the renal tubules and rest 1% comes out as urine. So the correct answer is 99%. This is value, this is digit. That's a direct answer. Just remember it. The next question is question number 9, which states that which of the following substances is not secreted by the tubular cells during urine formation. So during urine formation, which is not secreted by tubular cells. So we have to leave the substances that are being absorbed or secreted. We have to pay attention to it. So the very first option is sodium. You can't find it is NaCl or water or ammonia, but not this. So this is not secreted. So this is the answer. Other options like hydrogen, potassium and ammonium, you'll find they are secreted here. Ammonia, hydrogen, potassium, hydrogen, they are being secreted by the tubular cells. So these are the secreted products or substances. These are the reabsorbed substances here. So the correct answer to this question is option number A. Sodium ions are not secreted by the tubular cells during urine formation. Let's move on to the next question, question number 10, which is a lengthy question, so pay attention to it. The question states that read the following statements and select the correct option among the given options here. The very first statement states the descending limb of loop of Henle is permeable to NaCl and urea. The ascending limb of loop of Henle is permeable to water. So this statement is absolutely incorrect, because the descending limb of loop of Henle is permeable to water and not NaCl and urea. The second statement says that the osmolarity of medullary interstitium is of the order 1200 milliosmol per liter owing to the high glomerular filtration rate. So this statement again is incorrect, because the osmolarity of the medullary, the cortical medullary interstitium, it is owing to high glomerular filtration rate. It's in a gradient. It starts from 300 milliosmol per liter to 1200 milliosmol deep into the interstitium, and not this. The next is, third statement says angiotensin 1 is a powerful vasoconstrictor and increases the glomerular blood pressure and therefore GFR. This is incorrect, because it is not angiotensin 1 but angiotensin 2 that performs the function of increasing the GFR, right? The next question is, the next statement says atrial natriuretic factor or ANF, which is being released from the wall of heart during high blood pressure, it decreases the blood pressure by dilating the blood vessels, and this is a correct statement. So among the given statements, first is incorrect, second is incorrect, third is incorrect, fourth is the correct one. Let's see the options. The very first option say 1 and 2 are incorrect, 3 and 4 are correct. This cannot be a correct answer. 1 is incorrect and 2, 3, 4 are correct. This cannot be a correct answer. 1, 2 and 3 are correct and 4 is incorrect. This is not sure. This is incorrect. While the last option is 1, 2, 3 are incorrect, yes, and 4 is correct. So our correct answer to this question is option number D, that says the 3 statements are incorrect and the last one is the correct statement. So let's move on to the next question. Question number 11, again from the same chapter, excretory products and their elimination, and the question reads that read the following statements and select the correct option again. And the options are based on these 4 statements. The first statement says the porous cellophane membranes in a dialyzing unit allow flow of substance from blood to the dialyzing fluid against the concentration gradient. This is incorrect, because it is not against, but it is in a direction of this gradient. So it is based on the gradient difference and not against concentration gradient, right? So this is an incorrect statement. The next statement states that the glomerular filtration pressure GFP and capsular hydrostatic pressure CHP favor the glomerular filtration pressure, which is again incorrect. Let's go to this concept what it is. So whenever the filtrate has to be formed and filtration, ultrafiltration has to happen, there are certain forces that act upon it. The very first force that we are seeing in the question also, which is glomerular filtration pressure that is being created here, is based on this. So this is glomerular filtration pressure that supports the filtration. So it will be in a direction to support ultrafiltration. There would be glomerular hydrostatic pressure again in the direction to support it. And then there is colloid solute or capsular hydrostatic pressure that will oppose ultrafiltration, right? So in the question, the statement saying capsular hydrostatic pressure favor it? No, it doesn't favor it, it opposes it. So this statement is incorrect. The next statement says blood colloidal osmotic pressure BCOP resists the glomerular filtration pressure. So this statement is correct, as we have just seen that this colloidal pressure, which is because of capsular hydrostatic pressure, opposes and resists glomerular filtration pressure. And then you have the last statement, the counter current mechanism helps in concentrating the urine. This is also a correct statement. So if you go by this, the correct answer for this question would be option number B, that says 1 and 2 are incorrect, whereas 3 and 4 are correct statements. So the answer would be option number B. Let's move on to the next question, which is question number 12, that says that select the odd one with respect to flagellar movements. The very first movement, it helps in swimming of spermatozoa, is correct. In humans, spermatozoa exhibits flagellar movements. The second option, it helps in intracellular digestion in Euplectilla, which is a sponge, and sponges are characterized by the presence of choanocytes or collar cells. Which are flagellated cells, so they exhibit flagellated movements for the direction, for the movement of water in the body. So this is also correct with respect to flagellar movements. The third is helps in locomotion in euglena. Euglena is a protist that also show movement because of this flagellas on its body, and so it also exhibits flagellar movement. And the last is it is vital for the movement of secondary oocyte in fallopian tubule. This is incorrect and is the correct answer because it requires cilia, not flagella, and exhibits ciliary movement. We know fallopian tube is lined by ciliated epithelium to move the ova in one direction, in unidirection. So it is lined by cilia and not flagella, and the correct answer for this question is option number D. This is the odd one with respect to the events that is happening because of flagellar movements. Let's move on to the next question, which is question number 13, stating select the tissue sample in which we will find fusiform and uninucleate muscle fibers. So first of all, understand and identify the muscle fiber we are talking about in this question. The question states fusiform and uninucleate muscle fibers. That means we are talking about smooth muscle fibers. Smooth muscle fibers which are involuntary, uninucleated, fusiform and unbranched. And these are usually present in the hollow visceral organs. So the very first is ventricular wall. It comprises of cardiac muscle fibers. So this cannot be a correct answer. Second is tunica media of coronary artery, and this is the correct answer because coronary artery, the tunica media layer of coronary artery comprises of smooth muscle fibers. So this is the correct answer. The third is quadriceps muscle. It is a skeletal muscle. So this cannot be a correct answer. And the last is bicep muscles of upper arm, is also an example of skeletal muscle fiber. So this cannot be a correct answer. So the correct answer would be option number B, tunica media of coronary artery. Let's go to the next question, which is question number 14, and it is from the chapter Locomotion and Movement. The question states select the correct option among the following. And the very first statement is the Z line bisects the A band. Now, if you look at the sarcomere, the structural unit and functional unit of muscle fiber, you will see the sarcomere comprises of A band and I band. It comprises of myosin and actin filaments, the thick and thin filaments, and in between them you have this H zone. The Z line is here, that is present at the ends of one sarcomere. And the question is that Z line bisects A band. A band is here, Z line is here. So this is an incorrect statement. The next is myosin is thinner than actin. It is absolutely incorrect. Myosin is the thick band and actin forms the thin band. So this is incorrect. Third is sarcomere is the distance between two successive M lines. This is incorrect. It is the successive units. Sarcomere is this. It is not between two successive M lines, but it is between successive Z lines. And the last statement is the thin filaments are firmly attached to the Z line. This is correct. Thin filament actin, they are firmly attached to the Z line. So the correct answer, the correct statement in this, in these options given in this question is option number D. The thin filaments are firmly attached to the Z line, and so this is the correct answer. Let's move on to the next question of this paper, which is question number 15, stating axial skeleton comprises of how many bones? A direct question. We know that axial skeleton in human body comprises of 80 bones. So the correct answer is option number 2, not 120, 126 or 84. Let's go to the next question, which is question number 16 from the same chapter, locomotion and movement. And it's a fill in the blank kind of a question that states dash are considered false or vertebrochondral ribs. Vertebrochondral. This gives you a hint what type of ribs these are. That means chondral, that means some cartilage is associated with these ribs, right? So that means these are the ribs that are not directly attached to the sternum, but with the help of a cartilage, they attach with the 7th rib and then they form this association. So these are vertebrochondral ribs, and these are false ribs as they are not directly attaching with the sternum on the ventral side. Sternum is on the ventral side, vertebral column is on the dorsal side. And dash are considered true ribs. This is, we know that 1 to 7th pair of ribs are called true ribs because they are showing direct attachment to the sternum on the ventral side. So who are considered vertebrochondral ribs? As you can see here, these are the true ribs which are from 1 to 7th pairs. Then false ribs are vertebrochondral ribs which are from 8th to 10th pair, and the last 11th to 12th pair are called floating ribs. They are not attached to any other structure on the ventral side. They are just attached with the vertebral column on the dorsal side. So the correct answer is, who are considered as the vertebrochondral ribs? 8 to 9 pair? No. First 7 pairs and 8, 9 pairs? No, this is incorrect. 8, 9, 10 pairs are vertebrochondral and first 7 pairs are true ribs, so this is the correct option. 11, 12 pairs are floating ribs, so this is an incorrect option. So the correct answer is option number C. Let's go to the next question of this paper, which is question number 17, and the question states the coxal bone. The keyword is the coxal bone forms a part of which of the following? So if you see the hind limb of a human being, you will see here coxal bone. Coxal bone is a part of girdle in the hind limb of human. So are they associated with forelimb? No, coxal bone are not associated with forelimb. They are associated with the hind limb. They are associated with hind limbs. No, they are associated with girdle. So this is incorrect. They are associated with pectoral girdle. No, this is incorrect. They are associated with pelvic girdle is the correct answer. So these are coxal bone that forms the pelvic girdle. Pectoral girdle is formed of scapula and clavicle, right? So I hope this is understood now. Let's go to the next question, which is question number 18, that states about the disease which is formed because of low calcium in body fluids. So the question is, low calcium in body fluids causes which of the following disorders of the muscular system? And the question gives you the options like tetany, myasthenia gravis, muscular dystrophy and gout. So among the given options, the answer is option number 1, which is tetany. Please understand, this is not tetanus. Students often get confused with the option tetanus. So tetanus is a bacterial disease. Right, it's a bacterial disease, not because of low calcium in body fluid. It is caused by Clostridium tetani, right? And it causes, this is tetanus that causes it. Tetany is because of low calcium in the body fluid, which is a disorder of muscular system. The next is myasthenia gravis and muscular dystrophy. These are incorrect options. The myasthenia gravis is an autoimmune disorder. While your muscular dystrophy is a genetic disorder. These are the keywords that may come in your next NEET examination, so pay attention to it. And last is your gout, that is inflammation of the joints. So the correct answer is option number 1, that is tetany is the correct answer. Let's move to the next question, which states, question number 19, the saddle joint is formed between which of the following structures, and the options given are atlas and axis. This is incorrect because pivot joint is present over here, which is again a type of synovial joint. Next is carpal and metacarpal of human thumb. This is the correct answer where saddle joint is present. The next is between tarsals. This is incorrect. Here also synovial joint is present. Humerus and radius is also an incorrect option. It is also synovial joint but not saddle joint. So the correct answer is option number B, where saddle joint is present in humans. Let's move to the next question, which is question number 20. And again, as you can see, it's a difficult question. So let's move slowly over here. The statement is, read the following statements and select the correct option. The very first statement says, flooding of sarcoplasm with calcium occurs when sarcolemma gets polarized. This is important. It gets polarized or depolarized? When it gets depolarized. Action potential is formed when it gets depolarized on arrival of an action potential via a motor neuron. So this is an incorrect statement. It's a false statement. Next statement says, troponin and tropomyosin mask the actin binding site on myosin. This is incorrect because they mask myosin binding site on actin. Always pay attention to this kind of language. It is tricky and it comes in NEET, right? Tricky question comes in NEET. So always pay attention to where the site is. It is on actin to inhibit it of forming cross bridge to the myosin. So the sites are not on myosin. Myosin merely have sites for ATP binding and the site for actin binding. Whereas troponin and tropomyosin, they mask the myosin binding site which are present on actin. Okay. The next statement says, cross bridge between actin and myosin occurs when ATP detaches from the ATPase site on myosin head. So see, this is a correct statement because until and unless this ATP detaches from the ATPase head of the myosin and leaves the site for the attachment of actin, the cross bridge formation is not possible. So cross bridge happens when ATP detaches from it, and the energy this myosin will get from this ATP is used for this cross bridge formation. The next statement says, energy released due to ATP hydrolysis. This is what I'm talking about here is. So the energy released from this, it causes the actin filaments to be pulled towards the center of A band, and this is also a correct statement. So 2 statements are incorrect, 2 statements are correct. Which statements 1 and 2 are incorrect, whereas 3 and 4 are correct. Let's see which option justifies it. The very first option say 1 and 2 are incorrect and 3, 4 are correct. So this is the correct option. These rest of the options are incorrect. Let's move to the next question, which is question number 21, stating the 2 halves of the pelvic girdle. We have just seen pelvic girdle that comprises of coxal bones. The 2 halves of the pelvic girdle, they meet dash to form the pubic symphysis that contains dash cartilage. So the nature of the cartilage which is present here is fibrous. So this is fibrous cartilage, and they met ventrally, not dorsally. So the option that satisfies These fill in the blanks in the question is dorsally and hyaline is incorrect. Anterior and fibrous is incorrect. It's not anterior, it is ventral. Posteriorly elastic is incorrect because it's a fibrous joint and they are joined ventrally. So the correct answer is option number D. Let's move to the next question, which is question number 22, stating identify the correct statement among the following. And the very first statement says, Neissel's granules are found in cyton, dendrites and axon. No, this is incorrect because Neissel granules are not found in axon. The next statement is, neurons with 2 or more dendrites form the multipolar neuron is the correct statement. So this is the correct answer. Let's see the next statements also. Electrical synapses are common in our system. This is incorrect. They are rare in our system. It is the chemical synapses which are common in our system. The outer layer of cranial meninges is the pia mater. This is incorrect because it is the innermost meninge. So, the correct answer to this question is statement number B. Let's move on to the next question, which is question number 23. The statement is, the question is, the cerebral aqueduct, it's a ventricle that passes through which of the following parts of brain. So there are ventricles which are present in the brain and there are certain ducts that actually join these ventricles. Cerebral aqueduct is such duct which is also known as aqueduct of Sylvius or Iter. Let's see what it does. So as you can see here, this is the structure of a brain which comprises of these first and 2 ventricle. Then it forms this third ventricle which is connected by this. And then it forms this last ventricle which is connected by this cerebral aqueduct or aqueduct of Sylvius. And this is present in the midbrain. Your first and second ventricles are present in the forebrain, and then you have your cerebral aqueduct or aqueduct of Sylvius, which is in the midbrain. So your first option is incorrect. Here first and second ventricles are there. Hindbrain is incorrect. Spinal cord is absolutely incorrect. No such ventricles are found in spinal cord. They are in brain, and midbrain is the correct answer to this question. Let's move on to the next question, which is question number 24, stating which of the following is not a part of hindbrain. So hindbrain comprises of pons, medulla and cerebellum. So anything apart from it is not a part of hindbrain. Let's see the options. Pons, it's a part of hindbrain, so cannot be a correct answer. Hypothalamus is the correct answer as it is a part of forebrain. Then you have cerebellum, which is a part of hindbrain, so cannot be a correct answer. Medulla is also a part of hindbrain, so cannot be a correct answer. The answer is option number B. Let's see the next question, which is question number 25. It is about knee-jerk reflex. The knee-jerk reflex is a type of. So if you see this diagram, which is given in your NCERT, you will see here is the spindle fiber, the receptor over which when the sensation comes, this input is carried via efferent pathway through dorsal root ganglion into the spinal cord, and then through motor neuron it reaches to a site where motor end plate is there to give the response. And this type of reflex is known as monosynaptic reflex. So knee-jerk reflex is a type of polysynaptic reflex. No, it's a type of bisynaptic reflex. No, it's a type of trisynaptic reflex. No, it's a type of monosynaptic reflex. So the answer is option number D. Let's move on to the next question, which is question number 26, stating about the vestibular apparatus, that where it is located. So the vestibular apparatus is located in the dash ear dash the cochlea. So that's a direct line from your NCERT, very easy question, that your vestibular apparatus is located in the inner ear and is situated below cochlea. So the correct answer, above cochlea. It is situated above cochlea. Let's see the options. The very first option says it is located in the outer ear. No, outer ear doesn't comprise of this vestibular apparatus. It is to maintain the balance of the body. So it is not situated there. It is present in the inner ear, yes, but above the cochlea, yes. So this is the correct answer. It is situated in the middle ear. No, middle ear comprises of ear ossicles, the bones. And below the cochlea, this is incorrect. And the last is it is present in inner ear. Yes, it is situated in the inner ear, but not below cochlea. It is situated above the cochlea. So the correct answer is option number B. Let's move on to the next question. Question number 27. The question is, the reasoners and basilar membrane line the dash and dash respectively. So let us just see where these reasoners and basilar membrane actually situated. This diagram is given in your NCERT book. Pay attention to it. It has been asked once in NEET. So, excuse me. Where is the question? We are at question number here 27, right? So if you look at this diagram, which is given in your NCERT and has been asked in previous year question in NEET, you will see here these are the compartments which are formed because of these layers in the cochlea. This is your reasoner's membrane and this is your basilar membrane. So this basilar membrane which is forming the floor, and this is the reasoner's membrane that is forming the roof. Roof of what? Media. So roof of scala media and the floor of it, which is formed by basilar membrane. So this reasoner's membrane and basilar membrane, what it does reasoner's membrane forms the floor of the scala vestibuli and it forms the roof of the scala tympani. So they are associated with which of the these structures? So reasoners and basilar membrane, they line the eustachian tube and tympanum. This is incorrect. They line tympanum and eustachian tube. Absolutely incorrect. Eustachian tube connects your pharynx and oral cavity. They are associated with scala tympani and scala vestibule. No. They are associated with scala vestibuli and scala tympani respectively. So reasoner's membrane is associated with scala vestibuli and basilar's membrane is associated with scala tympani. So the correct answer is option number D. Let's move on to the next question, which is question number 28, which is about the layers in the retina. So arrange the 3 layers of neural cells in the retina from inside to outside. Let's have a very shortcut to remember it, which is, have you seen the movie Gangs of Wasseypur? Gangs of Wasseypur, GBP. This is the lineage. This is important. So if not, at least remember the name of the movie. That will help you with this. So if you go by this GBP, you will have the answer as ganglionic, bipolar and phosphoresis receptor, which is the correct lineage or arrangement of these layers in the retina from inside to outside. So the answer is option number D. Rest of the options is ganglion, photoreceptor, bipolar, incorrect. Photoreceptor, ganglion, bipolar, incorrect. The middle one is a bipolar. Then bipolar, ganglion, photoreceptor is again incorrect. The answer is option number D. Let's move on to the next question, which is question number 29. Select the correct statement among the following, and the very first statement says that in human eye, there are 3 different types of cones. And this is true, right? Let's see the other part of this statement. That contain photopigments responding yellow, red and blue light. See, the minute thing that make this statement incorrect is, it is not yellow, red and blue, but it is red, blue and green. These are the 3 different type of cones, photopigments in 3 different type of cones, and not yellow, red and blue light. So this make this statement incorrect. The next statement is at the anterior pole. The keyword is anterior pole of the eye, lateral to the blind spot, there is a yellow spot called macula lutea. This is correct, but the location is incorrect because it is at the posterior pole of the eye. This macula lutea is there, not at the anterior pole. This makes this statement incorrect. The next statement says the space between the lens and cornea is called aqueous chamber. Yes, it is true. The other half says contains a thin gel-like substance called aqueous humor. This is incorrect because it contains watery and not jelly-like substance. So this makes this statement incorrect. The last statement says the space between lens and retina is called vitreous chamber, which is correct, and contain a transparent gel called vitreous humor is a correct statement. So this is the correct answer. You can see here in the diagram of this eye, the sensory structure given in your NCERT 2, the structure which is situated between cornea and lens forms this aqueous chamber filled with watery fluid in this called aqueous humor, and between lens and retina is your vitreous chamber which is filled with jelly-like transparent material called vitreous humor. So the answer is statement D. Let's move to the next question, which is question number 30, which is about, this is a fill in the blank. So fill in the blanks with the appropriate option. The statement says the vibrations are passed through dash on the fluid of the cochlea, which induce a ripple in the dash. So you know the ripple is produced in the basilar membrane. That helps to understand there is difference in different pitches, right? So here would be basilar membrane, and it is through oval window. So your ear, middle ear that has stapes, the ear ossicle, that is in attachment with this oval window that lead it to the inner ear, and it causes ripple in the basilar membrane. Let's see the option that satisfies this fill in the blank. The very first is round window and scala vestibuli, which is incorrect. Second is the correct answer, which is stating oval window and basilar membrane. Third is round window, this is incorrect, and basilar membrane, so this is an incorrect option. The last is oval window and scala vestibuli is also an incorrect option. Let's move on to the next question, question number 31. Simple question that says that fibers of tracts are covered by. Very easy. So fibers of tracts are covered by myelin sheath is the correct answer. As you can see here, this is a nerve fiber, and this nerve fiber is covered by Schwann cells that form myelin sheath around it. This is the myelin sheath, and in between them there are nodes of Ranvier. So nodes of Ranvier are the places where this myelin sheath is absent. So the second statement, nodes of Ranvier, this is incorrect because these fibers of tracts are not covered with nodes of Ranvier, but these are the gaps in between two myelin sheaths. Third and fourth option, astrocytes and macrocytes, are incorrect. These are neuroglial cells. So the correct answer is option number A. Let's move on to the next question, question number 32, which says that excitement, pleasure, rage, fear, motivation is regulated by. And all these are regulated by our limbic system. Specifically, the things which are given here, they are regulated by amygdala. Amygdala. So you have hippocampus, you have amygdala, you have hypothalamus that constitutes the entire limbic system or limbic lobe, among which these emotions are controlled by amygdala. So the very first option, hypothalamus, is incorrect. Parietal lobe, which is the lobe present in the forebrain, is incorrect. Corpus callosum is incorrect. Amygdala is the correct answer to this question. Let's move on to the next question, which is question number 33, which is from the chapter Neural Control and Coordination, and the question is, read the following statements and select the correct option. As it's a long question, a difficult one, so we'll go slow with it. The very first statement says, at rest, the outer surface of axonal membrane is positively charged owing to comparatively higher permeability of the axon to potassium and near impermeability to sodium. So, this statement is correct. It is so. At rest, when the axon membrane is resting, it is in a polarized state. Then the outer axonal charge is positively charged, right? The membrane is positively charged because of impermeability to sodium and permeability to potassium ions. The next statement is, the opening of potassium pump restores the polarized state of the axonal membrane. And this is also true because we know from the polarized state it will undergo depolarization and again will undergo and reaches the polarized state because of the opening of sodium ions. So depolarization happened because of the influx of sodium ions, whereas again they will reach to the polarized state with the help of opening of the potassium channels. So this is also a correct statement that opening of potassium pump restores the polarized state of the axonal membrane from the depolarized state. Third statement says the sodium potassium pump remains operative only during depolarization is incorrect because it is functional even during this when hyperpolarization to polarization state is to be attained. So this is an incorrect statement. The last one is the relative concentration of sodium and potassium across the axonal membrane is restored after repolarization before turning off of the sodium potassium pump, which is incorrect because this pump is essential to restore this membrane potential. So this is an incorrect statement. So among the given options, 1 and 2 statement is correct, 3 and 4 are incorrect. So let's see the option that satisfies this. The very first is 1, 2, 3 are correct, 4 is incorrect, no. 1 and 2 are incorrect, 3 and 4 are correct, this is incorrect. The C is the correct answer, 1 and 2 are correct, 3 and 4 are incorrect, this is the correct answer. And the last one is also an incorrect option. Let's move to the next question, question number 34. From the sensory structures, at the base of cochlea, the scala vestibuli ends at the dash while scala tympani ends at the dash which opens into dash. So this is about the windows and the respective ears. Students often get confused on this indoors whether to answer oval window or round window. So there is a shortcut to remember it. The answer is depending on the shapes. So whenever you say stapes, stape opens through oval window into the inner ear, and that stapes has a oval kind of shape. So oval window comes first, that through which it enters into the ear, right, and rest would be your round window. So the very first is at the base of cochlea, the scala vestibuli ends at the oval window, while the scala tympani, then next would be a round window obviously. And they open into which ear? They open into middle ear, as stapes or ear ossicles, they are situated in the middle ear. So the correct answer that satisfies this fill in the blanks would be option number D, fenestra ovalis or oval window, fenestra rotunda or round window, and middle ear. Fenestra is the openings and it is ovalis because it is oval, and rotunda because it is round. The rest of the options are incorrect as they do not justify and satisfy the blanks given in the statement of the question. So let's move on to the next question, question number 35. Select the correct option or correct statement. The very first statement says the anterior pituitary is under the direct neural regulation of the hypothalamus. So we know that there is a connection between hypothalamus and pituitary gland. The anterior pituitary receives portal connection or portal circulation from hypothalamus, and it is not under direct neural regulation. It is posterior pituitary that is under neural regulation of hypothalamus. This hypothalamus will get a signal and it synthesizes oxytocin and ADH, which gets stored in posterior pituitary and get released from the posterior pituitary. Posterior pituitary do not synthesize any of its own hormone. They just stores the hypothalamic hormones. The second statement is somatostatin is secreted by the anterior pituitary. This is also incorrect. It is secreted from hypothalamus. GnRH regulates the synthesis and secretion of hormones from posterior lobe of pituitary. This is incorrect because there would be no synthesis, only secretion, only release of hormone from posterior pituitary, but not any synthesis of hormone. The last statement is the hormone responsible for the secretion and diffusion of pigment granules in skin is secreted from pars intermedia, and the hormone is known as MSH, melanocyte secreting hormone. So this is a correct statement and is the correct answer to our question. Let's move on to the next question, question number 36 of the section B. So section B comprises of the questions where you can choose. So there would be optional questions in this section. So let's discuss the very first question in section B. The question says the large bilobed glands located posterior to the vocal cords are. Options are parathyroid, thyroid, thymus and tonsils. So both parathyroid and thyroid, they both are situated posterior to vocal cords, but the bilobed structure is your thyroid gland. So this is the correct answer. Parathyroid is incorrect, thymus is incorrect, tonsils are incorrect. That's an easy question just based on the location of these glands in the human body. The next question is question number 37 from section B states, which of the following glands is responsible for secreting the hormone responsible for maintaining 24-hour diurnal rhythm of the body? Very important question, has been asked in NEET maybe 2-3 times in form of statement or just like that. So this is important. Who maintains 24-hour diurnal rhythm of the body? There is another question that could be framed, so please note that also. So one is your hypothalamus. That maintains the biological clock. And if this is not given in the options, then please go for pineal gland. This is a master clock, known as the master clock. This is given in your NCERT exercise. Right. So the correct answer is, if hypothalamus is not given, then pineal gland. First option pituitary, second thyroid, third is pineal, last is thymus. So the answer is pineal gland, as hypothalamus is not given in the option. Otherwise, hypothalamus would be the answer. So these are incorrect options and the answer is option C. Let's move on to the next question, which is question number 38, which is match the following columns and choose the correct option. In column 1, T4, PTH, GnRH and LH hormones are given. In column 2, the glands are given like hypothalamus, thyroid, pituitary and parathyroid. Let's match them. So T4 is released from thyroid glands. So 1 matches with 2. Then your PTH is released from parathyroid glands. So 2 matches with 4. GnRH is released from your hypothalamus, so 3 matches with 1, while LH, Leydig hormone, is released from pituitary, anterior pituitary specifically, so this 4 matches with 3. So any option that satisfies this is the correct answer. Let's see the option. The option is option number B, 1 with 2, 2 with 4, 3 with 1 and 4 with 3. So this is the correct answer. Let's move on to the next question. Question number 39. The dash hormone is hypercalcemic and hyperphosphatemic, while dash is hypercalcemic and hypophosphatemic. And the options are PTH and thyrocalcitonin, estrogen and thyrocalcitonin, PTH, PTH and adrenaline. So the shortcut to answer this question is to identify the hormones which are antagonistic to each other. And among them, antagonistic with respect to being hypercalcemic and hypocalcemic. Just select one of the feature. So if you go by hypercalcemic and hyper and hypo, then this would be hypo. So the answer would be option number 1, because PTH and TCT, thyrocalcitonin from the thyroid gland, they act opposite, antagonistic to each other. And this hormone PTH is hypocalcemic and hypophosphatemic, while this TCT is hypo and hypophosphatemic. This is also hypo. So the correct answer is option number 1 and not estrogen, TCT and PTH and adrenaline, because they act antagonistic to each other. Now let's move on to this. It is hypo, it is hypo. Let's move on to the next question, question number 40. Which of the following hormones would not require second messenger to mediate its effect? So the concept here is any hormone which is steroid in nature will directly pass through the cell membrane and will form intracellular receptor complex and don't need any second messenger, whereas proteinaceous hormones and other hormones, they require second messenger to mediate its effects. So among the given options, insulin, epinephrine, T3 and estradiol is a steroid hormone, so it doesn't require second messenger. Otherwise, insulin, epinephrine, T3, they require second messenger to mediate its effect. So the answer would be this and not these options. Let's move on to the next question. Question number 41 that states a patient suffers from a condition associated with frequent urination, excessive thirst and fatigue. Identify the hormone that will help relieve the symptoms. Now don't get confused here, as the options given are glucagon, insulin, somatostatin and adrenaline. Now, frequent urination, excessive thirst and fatigue indicates what? It indicates diabetes, maybe. Diabetes, frequent urination is polyuria, right? And fatigue and thirst is there. So the hormone that is given should be insulin, not glucagon that act antagonistic to insulin. It is not somatostatin, not adrenaline, which is a catecholamine and an emergency hormone. So the correct answer is option number B, that is insulin, which is released from pancreas. Let's move on to the next question. Question number 42 that says that if a patient is suffering from anemia, very first case, and low blood pressure, second case, goes to a doctor. The doctor may want to examine one of the following parts of patient's body for a comprehensive analysis of both the conditions. So both the conditions are to be associated with that particular body part. Identify the correct option. Let's go one by one. The very first option is collecting duct and hypothalamus. No such organ collecting duct can affect this, because anemia is not associated with collecting duct. There is no hormone that is released from collecting duct to control the formation of RBCs. Second is glomerulus and neurohypophysis. This is incorrect. Glomerulus is not associated with forming RBCs. Third is PCT and adenohypophysis. This is also incorrect, because PCT doesn't release any hormone that controls the formation of RBCs. The last is modified smooth muscle cells of efferent arteriole, and this is the correct answer to our question, because kidney is responsible for releasing a hormone known as erythropoietin and a hormone known as ADH or vasopressin that will act upon it. So erythropoietin to regulate anemia, and its filtration will affect glomerular low blood pressure, as it also releases hormone renin. So these two hormones are associated with kidney. So don't get confused, because ADH, vasopressin acts on kidney. They are released from hypothalamus only. So these cannot be correct answers. Right, adenohypophysis is not correct answer, because it doesn't release ADH and vasopressin. Answer is modified smooth muscle cells of efferent arteriole, which is associated with the structure kidney that regulates the formation of RBCs by the hormone erythropoietin. Renin regulates the blood pressure in the body. So the correct answer is this and not rest of the options. Let's see the next question, question number 43 that says, arrange the layers of adrenal cortex from outside to inside. And the shortcut is GFR. Right? You have heard about glomerular filtration rate, na? So just understand GFR is the shortcut, GFR. So whenever you find this GFR, that would be the correct answer. So zona glomerulosa, zona fasciculata and zona reticularis is the correct answer when you arrange it from outside to inside in the layer of the adrenal cortex. So the correct answer is this and not rest of the options. Let's move on to the next question, question number 44 that says, a 4-year-old patient undergoes a tumor excision for a treatment of epilepsy for a tumor located in the forebrain. This is important. Now, while growing up, the patient suffers from chronic conditions of bedwetting, excessive thirst. 25 years later, the patient conceives and faces trouble during parturition. Which of the following parts of the body might have been invasively affected during the tumor excision surgery? So I hope this case is clear to you in order to answer this, because in the question, the tumor is being excised from a part of the forebrain which may have affected some hormone that have to do something with parturition. So you know which hormone is responsible for this? Either oxytocin or maybe estrogen, cortisol. These are the hormones that are there when parturition is to happen, that play a role in the neuroendocrine mechanism that affects the fetal ejection reflex and favors parturition. Among them, cortisol cannot be an answer because it was not released from the brain. Estrogen, oxytocin, estrogen is released from ovary, so this cannot be the hormone. Maybe oxytocin. So let's see what other functions. Bedwetting and excessive thirst. So let's see. That means something is being affected that not only affects the release of hormone but other hormone that will regulate something to do with this. So this hormone we all know is what? It is ADH and vasopressin. And now you have to make this connection that these two hormones, oxytocin and ADH, is released from which hormone? Which gland? So these are synthesized by hypothalamus and they are released by posterior pituitary. Am I clear? Let's go to the options. The very first is thyroid, which is incorrect. Second is ovaries. This is incorrect. The examiner wants you to get confused with estrogen, but this is not correct. It is not a part of forebrain. Next is neurohypophysis, which is a correct answer. The posterior pituitary is known as neurohypophysis, that leads to the release of oxytocin and ADH for the maintenance of this parturition function and function to maintain body fluid and blood pressure. Last is your thalamus. That is not a correct answer. So the correct answer is option number C. This is a very conceptual and difficult question beyond NCERT, but that makes a concept, right? So pay attention to it. Let's go to the next question, question number 45, that says, due to the development of the nodules of the thyroid, which of the following symptoms can be seen? So when this is formed, this is because of exophthalmic goiter. That happens due to hyperthyroidism. And this exophthalmic is because of protrusion of eyeballs, right? So the very first option is protrusion of eyeball is associated, is the symptom of exophthalmic goiter or hyperthyroidism. Deafmatism is not associated with this. Abnormal skin and low intelligent questions, these are associated with cretinism. That happen because of hypothyroidism. So the correct answer is option number 1 and not B, C and D. Let's move on to the next question, question number 46, that says, which of the following will not occur due to impaired functioning of alpha cells in pancreas? Now alpha cells in pancreas is responsible for the formation of glucagon, whereas beta cells forms insulin hormone, right? We all know pancreas is a heterocrine gland that comprises of both exocrine and endocrine part. The endocrine part releases hormones like glucagon and insulin that acts antagonistic to maintain glucose level in the body fluid. So if we are talking about what will not happen under the effect of glucagon, let's just read what the glucagon is actually responsible for. So this glucagon, which is a peptide hormone, plays important role in maintaining the normal blood glucose level. It mainly acts on liver cells and stimulates gluconeogenesis. So this is the first part, that it stimulates glucogenolysis. Glycogenolysis, lysis of the glycogen, resulting in increased blood sugar. So there would be high blood sugar, there would be low glycogen stored in the liver. It acts on liver hepatocytes. In addition, this hormone stimulates the process of gluconeogenesis, formation of glucose from non-carbohydrate sources, which also contributes to the hyperglycemia. So glucagon is a hyperglycemic hormone. It reduces the cellular glucose uptake and utilization. So we have seen the functions of it, which are given in the options, that Which is not a function of glucagon? Does it affect the rapid movement of glucose from blood to hepatocyte and adipocytes? Yes, it does. So this cannot be a correct answer. Does it causes glycogenolysis, breakdown of glycogen? Yes, it does. So this cannot be a correct answer. Does it slow down the glucose uptake by the body cells from body? Yes, it does. So this cannot be a correct answer. All of the above is, which of the following will not occur due to impaired functioning? So if glucagon is not there, anything will not happen, as these are the function of glucagon. So all of the above will not happen. So the correct answer is option D, right? Let's go to the next question. Question number 197, that says, identify the structures primarily responsible to maintain balance of the body in humans. Now for that, ear is responsible. And what else? Ear and hindbrain, your cerebellum, right? So first option is forebrain and midbrain. This is incorrect. Midbrain and hindbrain is incorrect. Ear, yes, but hindbrain. So this is the correct answer. Your vestibular apparatus in the ear and your hindbrain are responsible for maintaining the balance of the body. Rest is midbrain and ear, which is incorrect. So the correct answer is option number C. You can also write vestibular apparatus in ear. Sometimes the question is so twisted, they ask you, it is being asked in previous year question, that which of the part of human body is not responsible for hearing but for maintenance of the body part. So this vestibular apparatus is not for hearing purpose, it is for the maintenance of the body, right? Let's see the next question, question number 48. Knee-jerk reflex in humans does not include. So we have already discussed knee-jerk reflex in this paper, but this is apart from this. In that question we have discussed it is a monosynaptic reflex. Any monosynaptic reflex, they lack interneuron. Please pay attention to it. It has been asked in previous year question in NEET. Very important. The very first option, interneuron, so is the correct answer. Knee-jerk reflex being a monosynaptic reflex. Lack interneuron. Motor end plate would be there, dorsal root ganglions would be there, motor neuron would be there. In the system, there would be efferent pathway, dorsal root ganglia, there would be motor neuron, there would be motor end plate known as effector, there would be a response that is created against it. The stimulus received at the receptor end. So the correct answer is interneuron. Let's go to the next question, which is question number 49, that states, hormones secreted from a gland located on the dorsal side of forebrain involved in the regulation of pigmentation, sleep-wake cycle, menstrual cycle, etc. is. So these are the functions which is associated with the gland situated on the dorsal side of forebrain, and that gland is your pineal gland. And this pineal gland releases a hormone known as melatonin. Don't get confused with melanin, that's for the pigmentation. Melatonin. So the correct answer would be option number D, melatonin. Oxytocin is incorrect. This is synthesized from hypothalamus but released from posterior pituitary. MSH is incorrect. This is released from pars intermedia. Melanin is incorrect. This is pigmentation creating hormone in the body. So the correct answer would be option number D, melatonin. Let's see the last question of this section, section B, and the entire paper, which is question number 50 from the subject zoology. The last question says that glomerular filtration rate is not affected by which of the following? So we know it is affected by BCOP, it is affected by CHP, that affects glomerular filtration rate, and it is affected by GHP. So they affect it. So in the options, there is blood colloidal osmotic pressure that affects GFR, glomerular hydrostatic pressure affects GFR, capsular hydrostatic pressure also affects GFR, but not oxygen partial pressure. Such an obvious question, right? Very easy question. The last question is the easiest question. Oxygen partial pressure does not affect glomerular filtration rate. It affects the exchange of gases, right? So the correct answer is option number D. So with this question, I would like to end the discussion of zoology paper from this AIRTS minor test 2. Let's move on to the next subject or next discussion of this paper.
Hello everyone, how are you all students? Today we are going to discuss about the minor test 2 and here we are going to discuss about botany portion. Students, here is the details of the test which has started on the 28th of November and these are the details here. This is the schedule of the test which are going. Now here we are discussing about the minor test 2 which was done on 12th of December here. So let's get started with our first question. See here, all of the following organelles are the part of endomembrane system except. So here we have to discuss about endomembrane system. What is it? See students, endomembrane system, it involves the organelles which are involved in the synthesis, transport of proteins and fats. Simple. Proteins and fats. Their transport, their synthesis, their packaging or modifications, they are being done by the organelles which are involved in the endomembrane system. So we are having here ER, we are having the Golgi bodies. So we have to see which organelle is not the part of endomembrane system. First option we are having peroxisomes, lysosome, vacuole and endoplasmic reticulum. As you can see in this image, we are having here the organelle. We are having here ER. Golgi is also the part of this endomembrane system. I can write it here, endomembrane. So it is involving ER, vacuoles, Golgi, but it is not involving here peroxisome. Peroxisome, they are also the organelles, very important part of the cell. Obviously, they are having some enzymes there for the digestion, for the breaking down, basically metabolism of the substances. But these are not the part of endomembrane system here. So peroxisome is the correct option. It is not part of the endomembrane. Secondly, which of the following is not a feature of facilitated diffusion in plants? Remember students, we have seen the diffusion, simple kind of diffusion. Obviously, it is a passive kind of process. When we talk about the facilitated diffusion, here the proteins are involved. What kind of proteins? Carrier as well as channel proteins. They are facilitating here the movement of the substances in and out of the cell. In case of plants here we are talking. That is why we are calling them as facilitated diffusion. Facilitation, these proteins, they are doing the facilitations of carrying the molecules in and out of the cell. So we are having certain characteristics here. We have to see which is not the feature of facilitated kind of diffusion. First one, highly selective. Obviously here the proteins, they are involved and it is highly selective kind of diffusion we are having because some substances they will be moved out, some they will not be moved in and out of the cell. B, transport saturates. So we are having the saturation, as I have told you, carrier channel proteins, they are involved here. So saturation, it does occur. Selective saturation, these are the characteristics of facilitated diffusion. C, requires special membrane proteins. As I have told you here, carrier channel proteins, they are involved in facilitated diffusion. So obviously we are having special membrane proteins. D, saying that uphill transport. Remember students, when we are talking about the facilitated diffusion, here we are not talking about the active kind of transport. So uphill transport is not possible here. It is obviously a passive kind of transport. So it will be downhill movement here. And what we understand by downhill movement here, from higher concentration to lower concentration. This is the characteristic feature of facilitated diffusion, as it is passive kind of diffusion having the downhill movement here from higher concentration to the lower concentration, not vice versa as we have seen in the case of active transport. So here correct option we are having is D. It is not the characteristic feature of facilitated kind of diffusion. Third question here is, which of the following is not observed when a cell is placed in a hypertonic solution? What do we understand by this hypertonic solution? Means that solution is having high concentration of solutes. Solutes, they are in high concentration in this kind of solution. That is why we are calling it as hypertonic solution. Water moves out of the cell when we are keeping a cell inside a hypertonic solution. So obviously we are having the movement of water from the cell to the outside environment. B. Protoplast shrinks away from the cell. When we are having the movement of the water out of the cell, obviously the protoplast, it will shrink, as protoplast is having this water. Mainly the water content is there in the protoplast. So it will shrink away from the walls. C saying movement of water occurs from lower water potential to higher water potential. First see the fourth option here. Water is first lost from the cytoplasm and then from the vacuole. So here students, C option is correct, because we are having here the movement of water. It will be from high water potential to low water potential. That is down the concentration gradient, not against the concentration gradient here. So C is the correct option here. And for D, obviously it will be lost first from the cytoplasm and then from the vacuole. Vacuole is also having a large amount of water in it. So we are having C option is correct, because water movement will be not against the concentration gradient, it will be down the concentration gradient. As you can see here in this image, hypertonic solution, we are keeping these red blood cells here. The water content is lost from the cell and it is going towards the environment, out of the cell. And if we are talking about the plant cell here, cell body shrinks from cell wall. Obviously water is lost from cell. So cytoplasm will lose the water and vacuole will lose the water. So we are having shrinking of the cell body here. Next question here is, bivalents tend to separate from each other except at the site of crossing over during. So we are having certain stages here. Pachytene of prophase 1, B zygotine of prophase 2, C diplotin of prophase 1 and D diakinesis of prophase 2 here. So we have to see here in which stage bivalents, they are separating but not at the site of crossing over. So I am having few images here. See here in the leptotene we are having this condition. Zygotine and pachytene we are having synaptonemal complex, and in this stage we are calling as diplotene. Chiasma formation is happening. When we are talking about the chiasma formation, it is happening because bivalents, they are separating but they are joined at the site where crossing over has been occurred. So we are having here the diplotene phase in which this stage will occur. Diakinesis, we are having this phase and metaphase 1. We are having the arrangement of the chromosomes here on the in the middle of the cell. So we are having correct option as C. Diplotene of the prophase here, we are having the joining of the bivalents here only at the site where crossing over has been done. Another question here is statement A. Amides contain more nitrogen than the amino acids. When we are talking about the amides, amides, they are having 2 NH2 group. And when we are talking about the amino acids, they are having 1 NH2 group. So if the statement is saying that amides contain more nitrogen than the amino acids, this is correct. Statement B says that transport of asparagine and glutamine to other plant parts occur via sieve tube of phloem. This is the incorrect option because it is occurring through xylem. Asparagine and glutamine, they will be transported with the help of xylem, not with the help of phloem here. So we are having statement A which is correct, whereas statement B, it is incorrect here. So only statement A is correct here. This is the correct option. Sixth question is saying choose the incorrect statement from the following. A. Occurrence of polysome for translation of single mRNA into proteins in prokaryotes. This is happening. When we are talking about the polysome, students, when we are having ribosome which is attached with the RNA, so we are calling it as polysome. String of ribosome attached with the RNA forming polysome. This is happening in the process of translation in case of prokaryotes. So this is correct here. The plasma membrane of prokaryotes is structurally similar to eukaryotes. This is also the correct option here. C saying gas vacuoles do not occur in blue-green algae. When we are talking about the blue-green algae, it is cyanobacteria, a prokaryote. Gas vacuoles, they are present in the prokaryotes because they perform the function of buoyancy. In case of cyanobacteria also, they are present. So C option or C statement here is incorrect and we have to choose the incorrect one also. D saying surface structures present in a prokaryotic cell are flagella, pili and fimbriae. So this is also the correct statement here. So we are having the correct option C, which is the incorrect statement here. So C is incorrect statement. Gas vacuoles obviously helping in the buoyancy of the cell. They are present in the prokaryotes, and cyanobacteria or blue-green algae, it is a prokaryote here. Another question here. Carbon dioxide acceptor molecule in Calvin cycle of sugarcane plant is. See here students, we have to see carbon dioxide acceptor molecule and in case of sugarcane plants. So A saying OAA, oxaloacetate here, B PEP here, C rubisco and D RUBP. You have to understand here rubisco is an enzyme. So the correct option obviously it will be RUBP. If we see the cycle here, we are having the carbon fixation. Rubisco, it is combining with the carbon dioxide and we are having the formation of PGA here. So obviously we are having here the D correct option RUBP, which is the acceptor molecule in the case of Calvin cycle and in the case of sugarcane plant. Another question here is respiratory electron transport system in plant is located at in the A perinuclear space, B mitochondrial matrix, C outer mitochondrial membrane or inner mitochondrial membrane. So we have to see where electron transport system is present in plants. See here, I am having this representation of the ETS or ETC, electron transport chain or electron transport system. Where it is present? We are having this cytoplasm. We are having here the outer membrane, intermembrane space that is between the outer membrane and inner membrane here, and the presence of electron transport chain or ETS. Where it is present? It is present in the inner mitochondrial membrane. These are the membranes of the mitochondria. See, this is the representation of mitochondria. We are having here the outer membrane, inner membrane. So ETS or the ETC, it is present in the inner membrane. So correct option here is D. Inner mitochondrial membrane is having the ETC or the ETS, electron transport system. Next question here is choose incorrect statement with respect to glycolysis. We have to see incorrect with respect to the glycolysis. A option is saying occurs in cytoplasm of all living organisms. This is correct. B, partial oxidation of glucose takes place when we are talking about the glycolysis. So obviously here the partial oxidation is happening. C, PEP is the final product of glycolysis. This is the incorrect because we are having the formation of pyruvate. Pyruvate is the final product of glycolysis. We will see in the flow chart here. As you can see, little blurry, but we are having the final product of glycolysis. What is it? It is pyruvate. So PEP is not the final product here, pyruvate. We are having, when the glycolysis is happening, we are having the formation of pyruvate. D is saying common process between anaerobic and aerobic respiration. Obviously, the presence or absence of oxygen will decide whether the oxygen is absent or whether oxygen is present. It will decide whether after glycolysis aerobic respiration is happening or anaerobic respiration is happening. So third option is here correct. Not the PEP, pyruvate is the final product of glycolysis. 10th question is select the mismatched pair. A, amyloplast is having the carbohydrate storage. Remember students, these are all the types of plastids. Amyloplast, leucoplast, elioplast and aleuroplast. So we have to see which one is mismatched here. Amyloplast, it is having the carbohydrate storage. This is correct here. B, leucoplast, it is having the pigment storage. This is not the correct because pigment storage, we have seen that in chromoplast, we have seen in the chloroplast. Elioplast for oil and fat storage and aleuroplast for the protein storage. So leucoplast is for, see here, leucoplast is for synthesis of fatty acids and amino acids. So we can, it is written here leucoplast, they are not for the pigment storage, we are having for the pigment storage chloroplast. So here the correct option is B. This is the mismatched pair. Another question here is choose incorrect statement regarding the phase lasting more than 95% duration of cell cycle. See here students, we are having this representation of the cell cycle. We are having here the mitotic phase and this is the interphase. See here, major portion we are having of the interphase here. So obviously 95% of the phase in the cell cycle is this interphase. So we have to see incorrect statement with respect to interphase. A, it is also called resting phase. So this is somehow correct because basically the cell division is not happening here, interphase. That is why we are calling it as resting phase. B, in this phase cell growth and DNA replication occurs. See here, G2 phase, cell growth is happening and DNA synthesis. These are all divided in this interphase. We are having the G2 phase, S phase, G1 phase and another one G0 phase also there. So we are having here basically cell growth and DNA replication. No cell division occurs in this phase. This is also correct with respect to the interphase. D is saying it starts with karyokinesis and ends with cytokinesis. When we are talking about the karyokinesis, that is the nuclear division. And when we are saying cytokinesis, that is the cytoplasm is dividing. And this is the characteristic of our mitotic phase. So this is not correct for interphase. So we are having the option D as correct. Another question, which of the following stages are part of karyokinesis during equational division? Remember students, equational division. What do we understand by it? It is mitosis. Here we are talking about the mitosis. So it is asking that in a mitosis, when karyokinesis is happening, when the nuclear division is happening. In which phases we will see the nuclear division or the karyokinesis in mitosis. So I am having here the representation. See here, prophase, metaphase, anaphase, and after that telophase. In telophase, what we are having? I can say that here cytokinesis is happening because cytoplasm is dividing. So rest of the phases we can say that karyokinesis is happening in mitosis. That is prophase, metaphase and anaphase. So nuclear division is happening in these phases. So we are having the options prophase, lactotin, pachytin. Here we are talking about the mitosis. So this is not correct for it. Anaphase, zygote and telophase. Zygote again, it is not correct. Diplotin, pachytin and anaphase. And fourth is saying prophase, metaphase and anaphase. Here the karyokinesis or nuclear division will be seen with respect to the mitosis. So here D option is correct. Another question, decapitation is a very useful technique in hedge making because due to that we are having the options senescence of leaves could be prevented, the bolting effect is induced in the plants, the fruit ripening in plants could be promoted, and D is the effect of auxin is removed and growth of lateral buds is enhanced. When we are making the hedges, in parks you must have seen the boundaries of the park, they are having the hedges. So obviously the hedge making is being done when the plant it has been decapitated because the apical buds they will be having auxin. And when we are having the auxin, we will see the apical dominance. And when we are having the apical dominance, the growth of lateral buds, it will not be promoted because we are having the growth of apical bud only. See here, apical bud, it is growing because auxin here is present in the apical bud. Lateral bud growth, it has been inhibited because of this apical dominance. So when we are removing this apical bud, we are having the removal of the auxin because apical bud, it was having the auxin. When apical bud has been removed because of this process, decapitation, auxin has been removed. Now we cannot see the phenomenon of apical dominance. Lateral buds, they will grow and it will make the plant bushy. Plant will be bushy because of this decapitation. So we can say that D option here is correct. The effect of auxin is removed, apical bud is removed and growth of lateral bud, it will be enhanced, making the plant bushy and it will be useful in hedge making, making the plants dense. So we are having the correct option as D. Another question here is choose the odd one out with respect to the macronutrients. Remember students, we have studied about the macro and micronutrients. We are having the options here, carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium. They are all macronutrients here. C, sulfur, magnesium and iron here. See here. And D, calcium, magnesium and oxygen. So odd one out here, obviously the C option, We can recall this, what are the macronutrients and what are the micronutrients. See here, iron is one of the micronutrients. Carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, NPK, very important for the plant. Calcium, magnesium and sulfur, they are all macronutrients. Other than that, these are micronutrients. So we are having the correct option C because it is having here the micronutrient. Another question, statement A says that cyclic photophosphorylation occurs when only light of wavelength beyond 680 nanometers is available for excitation. This is correct because we are having here the 700 nanometer in the photosystem 1. Statement B is saying during the cyclic photophosphorylation, water splitting complex is associated only with photosystem 2. Here we are talking about the water splitting complex. Is it present in cyclic photophosphorylation? No. Splitting of water is happening in non-cyclic kind of photophosphorylation. So this is incorrect statement here. Statement A is correct only. So we are having the options, only statement A is correct. This is the correct option here. Basically, water splitting, it is not photolysis of water. We can say it is not happening in the cyclic kind of photophosphorylation. We can recall it, we are having these differences, cyclic and non-cyclic. See here, photosystem 1 is involved. Photosystem 2 as well, photosystem 1 involved in the non-cyclic kind of photophosphorylation. Now where the photolysis of water is happening? Splitting of water is happening in non-cyclic kind of photophosphorylation. That is why we are having the statement B, which is incorrect, which is saying that cyclic photophosphorylation, it is having the splitting of water or the photolysis of water. It is not happening there. So we are having here the A option correct. Only A statement here is correct. Another question, in eukaryotic cells, which of the following is the small structure which work like oars causing the movement of either the cell or the fluid surrounding the cell? So we are having the options fibrae, B pili, C flagella and D cilia. So this is the simple question here because we are having the cell. Here we are talking about the eukaryotic cells. So we are having here that fluid surrounding the cell, or it will be causing the movement here. So we are having cilia. So they are also present in the eukaryotic cell, but I am having here representation of the this cell which is having this cilia. So one of the protist, it is having these cilia which will cause the movement of the water around it. So we are having the correct option as D. Another question here is, select the incorrect statement regarding the chromatin material. A saying it is present in nucleoplasm, B it is stained with basic dyes, C it contains DNA, some RNA, histone and non-histone proteins, and D it is usually observed during the division phase of the cells. Here we are talking about the chromatin and how chromatin is being formed, DNA as well as the proteins. So C option here is, C statement here is correct obviously. It is stained with the basic dye, this is also correct. It is present in the nucleoplasm. So we are having the presence of chromatin material in the nucleoplasm also. But here D is incorrect and the correct option here. It is usually observed during the division phase of the cells. This is not correct. Initially we are having the presence of the chromatin. After that chromosome formation will happen. So D option here is correct. We are having this representation chromosome. In the later, when the division of the cell is happening, we will see these structures. In the initial phase, we will see here the chromatin fiber, and it will be formed of these RNA, histone, non-histone proteins as well as the DNA. So D is the correct option. Another question, splitting of centromere and separation of chromatids. It occurs during anaphase of mitosis and anaphase 1 of meiosis. Anaphase 1 of meiosis only, anaphase 2 and anaphase 1 of meiosis, and anaphase, we are having here the 3 anaphases, anaphase 1, 2 of the meiosis here and anaphase of the mitosis here. So we have to see where splitting of centromere and separation of chromatids is happening. Remember, separation of chromatids will be present where? I am having this representation. Here we are talking about the anaphase of the mitosis. Here we are having the anaphase 1 and here anaphase 2. See here students, where is the separation of chromatid is happening? It is saying splitting of the centromere and separation of the chromatid. It is happening in the anaphase of the mitosis. This is correct. Anaphase 1. Is it here happening the chromatid separation? No. See here the whole chromosome is there. No separation of the chromatid. Anaphase 2, we are having the separation of the chromatid. We are having here the chromatids. Splitting of the centromere and separation of the chromatid. So it is happening in the anaphase of the mitosis and anaphase 2 of the meiosis. So correct option here is D. Whereas in the anaphase 1, we are having the chromosomes on the respective poles here, they are going, but it is not happening here, the separation of the chromatid. Another question here is, the eukaryotic cell organelle involved in formation of glycoprotein and have all the features, functions except when we are having the formation of glycoprotein. When we were talking about the endomembrane system, what I have told you there? Modification, transport of lipids, proteins will happen. Endomembrane system, lipids, proteins, their modification, their transport will happen because of this endomembrane system. Here we are having the formation of the glycoprotein. So obviously it is a modified, we can say the protein, glycoprotein, it is carbohydrate plus protein. And which organelle is responsible for it? So the organelle which is responsible for it is Golgi apparatus. Which of the following are characteristics of this Golgi body and which is not? So A is saying having distinct concave and convex faces. So this is the characteristic feature of this Golgi apparatus we have seen. B is saying involved in the secretion of materials outside the cell. This is also happening when we were saying the modification, the formation and the transport of molecules, it will happen. Participates in oxidative phosphorylation. See students, when I was talking about photophosphorylation, we have seen that ETS, where it was present in the mitochondria, inner membrane of the mitochondria. It was having this ETC or ETS. So obviously oxidative phosphorylation is a function of mitochondria, not the Golgi body here. So obviously this is not the characteristic feature of this Golgi apparatus. D, remain in close association with ER. It is forming the endomembrane system, so it is associated with the ER, this Golgi apparatus. So this is also correct. So C option is correct option and the wrong statement for the Golgi apparatus. As you can see here, we are having this Golgi apparatus. Cisternae apart, transphase, cis phase is present, convex, concave phases they are present in association with the ER. These are all the characteristics of this Golgi body which is having the formation of glycoproteins. Another question, name the scientist who first described action spectrum of photosynthesis. See here we are having this action spectrum. What do we understand by it? Rate of photosynthesis will be higher at which wavelength? Which wavelength will be having the most effective photosynthesis? This is being explained by action spectrum. Wavelength basically in the blue, violet and red region. blue violet and red region will be having blue violet and red region will be having maximum photosynthesis. This is being explained by action spectrum and the experiment which was done by Engelmann. So we are having this representation of the Engelmann's experiment. We can summarize it. He took the prism and he did the splitting of the white light in the VIBGYOR here. So he was having this algae. It was kept in this splitted white light. We are having the VIBGYOR here. And the oxygenic bacteria which will go towards the oxygen for its metabolism, they were concentrated near the regions which were having blue violet and the red light. So we are having the aerobic bacteria. It is concentrated basically in these regions, in blue violet and red region. And this is explaining that photosynthesis rate is maximum in blue violet and red region with the help of algae, as algae is producing or evolving here the oxygen because of this photosynthetic process. So Engelmann's experiment was that, and we can say that scientist who first described this action spectrum where the photosynthesis will be maximum, it was done by Engelmann. So the A option here is correct. Another question, select the incorrect physiological effect of ethylene. The plant growth regulator here, that is gaseous in nature. Asynchronization of fruit set in pineapple, this is correct for ethylene. Promotion of female flower in cucumber. Thinning of cotton, cherry and walnut, and D, inhibition of flowering in mango. So students, D option here is incorrect. We have to see incorrect with respect to the ethylene. Why? Because it is not doing the inhibition of flowering. It is promoting flowering. As we can see here, the functions of this ethylene. Transfer growth, senescence. One thing you have to remember that ABA and ethylene, they are plant growth inhibitors. That is why we will see a lot of words like abscission, senescence in case of ABA and ethylene. So we are having the transfer growth here. Senescence, abscission, fruit ripening, one of the main function of this ethylene. Breaking of dormancy, elongation of aquatic forms, roots and root hair, flowering here. That is why the D option there is correct. Sex expression and ripened fruits here. So obviously D option here is correct because it is promoting flowering. Another question, meristematic cells have all the following characteristics except dense cytoplasm. You can see here this is the representation of the cells which are from meristem. So they are having this dense cytoplasm. This is correct for it. This is correct with respect to the meristematic cells. Small conspicuous nucleus. So the nucleus here, it is not small. So this is incorrect with respect to the meristematic cells. Thin cellulosic primary cell walls. As students, they are dividing, continuously dividing. That is why we can say they are having thin cellulosic cell walls here, continuously dividing. We are here talking about the meristematic cells. D, abundant plasmodesmatal connections. This is also the characteristic of this meristematic cells here. So we are having the correct option B. As you can see here, they are having the large nucleus. They are not having the small nucleus here. So this option B here is correct. Another question, during rainy season, wooden doors generally swell due to A, osmosis, B, facilitated diffusion, C, imbibition, and D, transpiration. As you can see here, students, in this image, in the first image, it was not kept in the water. That is why it is quite shrinked here. But after keeping it in water, it has absorbed the water. Its surface, it has done the adsorption there. And this has happened because of the process of imbibition. Adsorption of water on the surface is causing this imbibition here. So when we are having the wooden doors in the rainy season, they will swell. As the wood, it will absorb the water on its surface. So we are having the options osmosis, facilitated diffusion, imbibition and transpiration. So it is happening because of the process of imbibition here. C option is correct. Another question, statement A. Loss of water in its liquid phase through stomata is called guttation. Here the statement loss of water. In the guttation it is happening. In the liquid phase it is happening. But it is not happening through stomata. It is happening through hydrothodes. We are having the structure which we are calling as hydrothodes, which are doing this process of guttation, not the stomata. So statement A is incorrect. Statement B is saying root pressure is responsible for pulling water to small heights in the stem. So when we are having this statement, we are having here the pushing up of the water. This statement will be correct this way. So we are having the statement A and B both are incorrect. We are having the correct option here is D. Both statements A and B, they are incorrect. Remember students, root pressure, it will push up. It is not pulling here. That is why this statement is incorrect. As in the case of guttation we have seen, it is happening through hydrothodes, special structure for this process. See here. See here, we are having this guttation process and it is happening because of this hydrothodes. And when we are having the root pressure, it pushes water to the stem up to a small height. That is why we are saying that it is pushing up, not the pulling. Another question, select the mismatched pair here. We have to see the mismatched. A, abscisic acid is a stress hormone. GA, bolting effect, gibberellins, they are having these bolting effect. C, auxin, induction of parthenocarpy, and D saying cytokinin, that is the promoter of leaf senescence. This is the incorrect pair, or I can say mismatched pair here. That is why this option is correct. As we can see here, the uses and the functions of cytokinin. See here, is it promoting leaf senescence here? It is doing the delaying of the senescence. Other functions we can see, growth of the leaves, growth of the shoot, overcome apical dominance, delaying senescence. That is why that option is correct there. And cell division. So cytokinin, students, we can say that it is growth promoter hormone, not a growth inhibitor hormone. Another question, dense particles observed by George Palade, also called Palade particles, are composed of. When we are talking about the Palade particles or the particles which were discovered by George Palade, they are ribosomes. And when we study about the translation process, we have seen there ribosome, it is composed of the RNA as well as the proteins, amino acids there. So we have to see it is composed of, the options here are RNA only, this is not the correct option, RNA and DNA, RNA and proteins, and RNA, DNA and proteins. So C here is the correct option. It is having the RNA and proteins. If we visualize it, we are having these subunits and we are having here the RNA. We are having these We are having these subunits and this is composed of the whole structure of the ribosome, of the RNA and the protein. So here C option is correct. Another question, the outer membrane of nucleus usually remains continuation with dash and bears dash on it. A is saying Golgi apparatus and lysosomes. Here we are talking about the nucleus membrane, nuclear membrane. ER and the ribosomes, lysosomes and the ribosomes, and subvacuole and the tonoplast. If we see here, we are having this nuclear envelope. See here, nuclear envelope, and it is in continuation with endoplasmic reticulum. We can say smooth or rough endoplasmic reticulum. And this endoplasmic reticulum, that is, which one is rough endoplasmic reticulum, it is combined with the ribosomes. Ribosomes, they are attached on it. You can see here. So the option here will be correct is B. Nuclear envelope, it is in continuation with the endoplasmic reticulum, as we can see here, and it is studded with these ribosomal structures. Ribosome. Another question, identify true or false statement with respect to the electron transport system. Remember students, ETC or ETS here, where it was present in the inner mitochondrial membrane. As we have seen earlier also, we have to see which statement is correct for it and which one is false for it. First statement, ubiquinone receives reducing equivalence via complex 2. Second is saying cytochrome C is a big protein attached to the inner surface of outer mitochondrial membrane. Remember? And third is saying oxygen acts as the final electron acceptor. First we have to see what is happening here. See here, electron acceptor, oxygen, this is correct here. And it is saying that cytochrome C is a big protein and it is attached to the inner surface of outer mitochondrial membrane. So this is false here. It is all happening on the inner membrane. First is saying ubiquinone receives reducing equivalence via complex 2 here. See here, this is also correct. So it is having the interconnection here, ubiquinone, and we are having it is receiving through complex 2 here. This is also correct here. So we are having the correct option as B1. Second one is false. As we have seen, the inner mitochondrial membrane, we are having this ETC or ETS. Another question, which of the following PGR acts as an antagonist to gibberellins? We are having A, oxin, B, abscisic acid, C, ethylene, and D, cytokinin here. When we were talking about the antagonism, we have seen that gibberellins, they are antagonists to ABA. We have seen these are plant growth inhibitors. And these are plant growth promoters. We can also see it here. See here, gibberellins, stem growth, molting, seed germination. Whereas in the case of abscisic acid, abscission, dormancy of buds and seeds, a seed dormancy is happening here, where seed germination is happening in case of these gibberellins. Metabolism inhibition, inhibition of seed germination, direct antagonistic effect here, closure of stomata here. So we are having the opposites here, gibberellins and abscisic acid. So here B option is correct. Next question, if a pressure greater than atmospheric pressure is applied to pure water, its water potential. We know that water potential of pure water, it is 0. And when we are applying pressure greater than the atmospheric pressure, it will increase. It will not decrease, it will increase. So obviously we are having the correct option here is B. That is, it will increase as the water potential of the pure water, it is 0, and when we are applying the atmospheric pressure, it will increase here. Next question, apoplastic movement of water, it occurs through A, intercellular spaces, B, cell wall, and C, interconnected protoplast. See here students, in this image, we are having the symplast and the apoplast. Dark colored, it is the apoplast, where it is happening in the cell wall as well as in the intercellular spaces. So we are having symplast, that is the lighter one, through the inner region of the cell. So we can say that D option is correct here. Interconnected protoplasts, they are having this symplastic kind of pathway here. Another question, Transpiration increases with decrease in temperature, C saying available water for the plant, C is sunlight, and D humidity. So when we are having humid conditions, transpiration it will decrease. It is having alternative relationship with the humidity. As you can see here, when we are having the wind velocity on the plant transpiration, so it is having quite direct effect here. And when we are having the temperature on the plant transpiration, what is happening? It is getting accommodated, or it is having the stable condition here after some time. When we are increasing the temperature up to a limit, it will increase the transpiration, but after some point it will be getting stabilized here. In the effect of humidity, it is having indirect relationship with the transpiration. See here, rate of transpiration and humidity. When we are having less humidity, rate of transpiration will be increased. When we are having low humidity, it will be having the rate of transpiration, it will be higher. So we can say that transpiration, it increases with decrease in case of humidity. When we are saying about the sunlight, it is increasing up to a limit, but after that it is getting stabilized there. And when we are talking about the temperature, available water for the plant, it is not having this indirect relationship, whereas in the case of humidity, it is having this indirect relationship here. Another question, this is also the representation here of the transpiration having the relationship with the humidity. Transpiration, it is lower when we are having the high humidity in the outside environment. It is higher when we are having lower humidity in the outside environment. Another question here is, if an animal cell has 10 chromosomes and 20 pgDNA, when we are talking about the pg, that is the picogram, and it tells us about the mass of DNA. So students, it is saying that before the S phase, we are having this amount, 10 chromosomes and 20 pgDNA. What will be the number of chromosomes and amount of DNA respectively in the same cell after the S phase? After the S phase, what will happen? What will be the amount here of the DNA and the number of the chromosomes? When we are talking about the S phase, we are having there doubling of the DNA content. So we can say that 40 pg will be the DNA, and we will not have the effect on the chromosomes. It will be 10, only 10 chromosomes. Because we are having doubling of the DNA content, doubling of the DNA amount there. So we are having the correct option as D. S phase associated with this doubling of DNA content. Another question, which of the following is not an application of transpiration for plants? A. Water supply for photosynthesis. Transpirational pull we have seen. So this is also the effect of this transpiration on plants. B. Cooling of leaf surfaces. When water is getting outside the plant into the environment, we are having the cooling. Obviously, this is also the effect of this transpiration. C. Saying C. Maintenance of leaves in flaccid state. This is not correct statement here. You will see why. D. Saying transport of minerals from soil to different plant parts. So when we are having the transpirational pull, it is also having the conduction of water and minerals inside the plant. So this is also the correct statement here. Maintenance of leaves in flaccid state, this is not the correct option. See here students, turgid cells we are having in the plant. Transpirational pull will be having the turgidity, not the flaccidity here. This is the flaccid plant here. So this is not correct for the transpiration. We are having here this option as correct. Another question, How many of the following cell organelles are common between blue-green algae and red algae? When we are talking about the blue-green algae, it is a prokaryote here. And when we are talking about the red algae, it is the eukaryote here. So basically we have to see the difference between the prokaryote and eukaryote. Which kind of organelles? I cannot say that organelles because obviously the prokaryotes, they are not having the well-defined organelles there. So we are having here mitochondria, mesosome, chromatophore, nucleoid, Golgi body, lysosome, vacuole, ribosome, endoplasmic reticulum and nucleus. What are common in between the prokaryotes and the eukaryotes? So, eukaryote represented here with the red algae, and prokaryote, it is here being represented with the help of cyanobacteria. So we can have this comparison here. So what kind of organelles they are present? Organelles, or we can say the not well defined organelle is present in these. So see here, plasma membrane, common. Cytoplasm, you can see the structure of the cell, structural component of the cell. Ribosome, it is also present. Other organelles, as you can see here, they are not present in this prokaryote. And this cell wall capsule nucleoid is also not present in the eukaryote. So we are having these three similar structural components here. So obviously out of this, we are not having here the cell membrane and we are not having the cell wall. We are not talking here about the cytoplasm. What we are having similar here is ribosome. So I can say that only one of the following cell organelle, cell component here, it is common. So A option here is correct. Another question, identify the incorrect feature with respect to the guard cells of the leaves of dicot plants. So here we are talking about the dicot plants and we have to see the guard cells. What are the characteristics there? A, bean-shaped structure. This is correct for dicot plant. B, thick elastic inner wall. It is also having here in the dicot plants. C, longitudinally arranged cellulose microfibrils. This is incorrect here because they are radially arranged. D, occurrence of chloroplast. Chloroplast is also present in the guard cells. So we are having here the correct option C. As you can see here, we are having these guard cells in case of dicot plants. So they are bean shaped. As you can see here, they are having here thick and elastic inner wall. See here, thick here. Occurrence of chloroplast is also present. Chloroplast is also there. But are they longitudinally arranged? These microfibrils here, they are radially arranged. Radial arrangement. So we can say that the C option here is correct. Another question, select mismatch pair. A, negatively hydrostatic potential, water column in the xylem. B, positive hydrostatic pressure, it is root pressure. C, solute potential is always positive. D, water potential of pure water, it is always 0. So obviously students, all these three statements, they are correct except this one. This will be our correct option as we have to see the mismatched pair. Solute potential, when we are talking about, what is that? When we are adding solute in the water, we are having certain solute potential. It will be negative. It will not be positive here. As you can see here, it is a negative number. Solute molecules, they have been added in the pure water. It is decreasing the water potential here. So obviously this solute potential, it is a negative number. Semi-permeable membrane, as we can see, the pure water, it was having the water potential. This is 0. So correct option here is C. Solute potential, it will not be always positive, it will be negative. Another question, which of the following is non-membrane bound organelle, present in animal cells but absent in plant and bacterial cells? So we have to see non-membrane bound organelle which will be present in animal cells. Here we are not talking about the plant and bacterial cells. So ribosomes, are they present in the plant cell or bacterial cells? Obviously they are. B, nucleolus, it is present here. obviously it is also present. C. Centrosome. These are the these are mainly present in the animal cells. So C option we are having here correct. D. Mitochondria. Obviously the energy we are requiring, animal cell as well as plant cells they are requiring here. So the mitochondria is also present there, but the centrosome or the centroles, they are the characteristic feature of the animal cells. As you can see here, centrosomes, they are present in this animal cell. Another question, a cell organelle X is specifically stained with Janus green and its number per cell varies depending on physiological activity of the cell. So whenever we are staining this certain organelle, it is associated with some physiological activity. The organelle X here is, students, when we are talking about the Janus green, it turns green when it is oxidized. This is the basic mechanism or the phenomenon behind it, and it will turn that organelle where it is present. So mitochondria, it is doing this process, and we know that mitochondria, they are related to the physiological activity. So they are doing this oxidation of this Janus green, and when this Janus green is combining with this mitochondria, because of the process of oxidation, turning this mitochondria green. So we are having A. chloroplast, B. mitochondria, C. nucleus, and D. lysosome. So here correct option is mitochondria. Janus green is staining this mitochondria green, which is related with physiological activity. See here, these are the mitochondria. Another question, membrane-bound vesicular structure formed by the process of packaging in Golgi apparatus is A. Smooth endoplasmic reticulum, B. Rough endoplasmic reticulum, C. lysosome, and D. vacuole. So we have to see membrane-bound vesicular structure, and it is forming by packaging of Golgi apparatus. It is obviously the lysosomes here. They are having this formation because of the Golgi apparatus. As we can see here, lysosomes, because of the formation of these Golgi apparatus here, we are having the ER, we are having basically the part of the endomembrane system, and we are having the process of packaging of these lysosomes here in this Golgi apparatus. See here, this is also the structure of lysosome here. Glycosylated membrane, it is having the proteins, they are present containing these hydrolytic enzyme mixture here, lysosome basically. And it is all the part of modification, packaging, transport we are having through the endomembrane system. Another question here is, which of the following stages of karyokinesis shows the below mentioned event? When we are talking about the karyokinesis, we are having here nuclear division. And which of the following stages they are occurring in this karyokinesis? Chromosomes cluster at the opposite spindle poles. B saying nuclear envelope develops around the chromosome. Third is saying nucleolus, Golgi complex, and ER, they are reformed here. See here students, this is the stage here. We are having these characteristics, and we are all having here in this telophase. We are having here the membrane forming the function here. See. Nuclear division, nuclear envelope, it develops around the chromosome, and the chromosome cluster at the opposite spindle poles, and the nucleolus, Golgi complex, and ER reform. So it is all happening in this phase here. You can see here Golgi, ER, they are coming here, and in the telophase we are having here. After that, cytokinesis will happen. Another question, select the incorrect statement with respect to the reductional division. Here we are talking about the meiosis. Involves 2 sequential cycles of nuclear and cell division. B involves single cycle of DNA replication. Involves pairing of non-homologous chromosomes. This is the incorrect option here, because we are having the pairing of homologous chromosomes. Recombination between non-sister chromatids of homologous chromosomes, it occurs. So we are having here the correct option, because we have to find out the incorrect statement. As you can see here, we are having here the homologous chromosomes. Pairing of not non-homologous chromosomes, obviously pairing of the homologous chromosomes, it is happening here. Another question here is, we have to see the mismatched example of nitrogen fixing bacteria. Rhizobium, a heterotroph, symbiotic and present in the legumes here. So symbiotic association with the legumes. Frankia, heterotrophic, in association, symbiotic association with the non-legumes. And the third one, Anabina, it is heterotroph and symbiotic, free living, and is present in the non-legumes. This is not the correct matching here, because it is present in the Azolla, which is a fern, and we cannot say that it is a non-leguminous or leguminous plant. So this third one is the mismatched pair here. So we are having the correct option as B. Another question with reference to the factors affecting photosynthesis, which of the following statement is incorrect? A, dark reaction being enzymatic is temperature controlled. B, tomatoes and bell peppers show higher productivity. Tomatoes and bell peppers show higher productivity at higher carbon dioxide concentration. C, light saturation of the rate of photosynthesis occurs at 5% of full sunlight. And D, water stress causes stomata to close, hence reduces the CO2 availability. So students, in the third statement, we can say that, see here, the light saturation point. It will occur at 10% of the full sunlight. So obviously here, we have to see the incorrect statement. C1 is that. Because dark reaction, obviously enzymes are involved there and it is getting affected because of the temperature. Tomatoes and bell pepper, they are showing the increase in the photosynthesis when we are having the higher carbon dioxide concentration. Water stress, it will cause the stomata and we will have the shutting down of the transpiration. So all the 3 statements, they are correct, whereas the C1 is incorrect here. Which of the following is absent in maize plant but observed in tomato plant? Occurrence of Calvin cycle in mesophyll cell. B, photorespiration process. C, involvement of ribosco in primary carboxylation, all except C. So here students, we are talking about the C4 plant and here we are talking about the C3 plant. So we are having here the ribosco involved in the primary carboxylation. This is not true. As you can see here, we are having the involvement of the PEP in case of C4 plants. So obviously, we are having this correct for the tomato plant but not for the maize plant. So obviously, this one is correct option here. We can say all are correct, but C is not correct for the maize plant here. Another question, RQ value of protein as a respiratory substrate is 0.7, 0.91 and 1.5. When we are talking about the RQ value, it is respiratory quotient. So RQ value is equals to amount of CO2 evolved, volume I can write, and volume of the oxygen consumed. This is for the RQ, and for the proteins it is approximately 0.8, 0.9. So we can say that B is correct option with respect to the proteins, as we are having the respiratory question which is equal to volume of the CO2 evolved divided by volume of the oxygen consumed during the respiration. Another question, statement A says that plant growth regulators are small, simple molecules of diverse chemical composition. This is correct, obviously plant growth regulators. Statement B is saying ethylene is a gaseous PGR largely involved in growth promoting activities. In the beginning of this discussion, we have said that ethylene as well as abscisic acid, they are growth inhibiting. They are growth inhibiting regulators here. So obviously this is the incorrect option here, incorrect statement here. So only statement A is correct. Another question, as you can see here, ethylene, it is the plant growth inhibitor. Another question here is photoperiod is necessary for flowering induction and its signal is perceived by. We are having a hormone which is present and it is having the flowering induction and its signal is perceived by the leaves here. So the C option here is correct. Another question, experiment which demonstrated the source of auxin was a fungal extracts from rice seedlings. We have seen that this is for the gibberellins. Ripening of bananas from the volatile substance, obviously ethylene there. C, bending of the coleoptile tip of canary grass. This is correct for the auxin. As I've told you earlier also, when auxin is present in the apical buds, it is showing the apical dominance, and we have seen in this case of experiment also, when we were removing this apical bud, when we were decapitating the plant, we are not having the effects of the auxin because auxin is present there in the apical buds. So this experiment, it proved that where the source of auxin is present. Another question, statement A, diffusion in solid is more likely rather than of solids. So when we are talking about the diffusion, substances, molecules, they will go from higher concentration to lower concentration. So substances, solutes, they are having this diffusion, gaseous molecules also there. So A option is, A statement here is correct. B statement, phloem sap is mainly water and sucrose. So what kind of sugar we are having in the phloem sap? We are having here the sucrose and mainly the water here. So B statement here is correct. So we are having the correct option here is C, because both statements A, B, they are correct here. So students, today we have discussed about the botany part of the minor test 2. Keep learning.
Good afternoon, everyone. I will be discussing the AIRTS minor test 2 chemistry portion. This test was conducted on the 12th of December. The syllabus it had thermodynamics, equilibrium, states of matter, hydrogen, redox reactions, S-block elements, general organic chemistry, and hydrocarbon. So let's start with the questions. The first question was based on thermodynamics. Here the S0 value, that is the entropy values of hydrogen, was given as 0.22 kilojoule per Kelvin per mole. Cl2 was given as 0.13 kilojoule per Kelvin per mole. And the HCl value was given as 0.19 kilojoule per Kelvin per mole. They have asked you to calculate the total change in standard entropy. It's a very, very simple question. So the change in entropy will be equal to the sum of the entropy of the products minus the sum of the entropy of the reactants. So on the product side, we have 2 HCl. So we will be taking 2 times the value of HCl, that is 0.19 kilojoule per Kelvin per mole, minus the sum of the reactants. In the case of reactant, we have 0.22 plus 0.13. So if you solve it, you're going to get 0.38 minus 0.35, that is 0.03 kilojoule per Kelvin per mole. But the answer is given in joule per Kelvin per mole. So we will just multiply this answer by 10 to the power of 3 and we get 30 joule per Kelvin per mole. So C is the answer. So the next question is also based on thermodynamics. Here they have given the equation in which the water in the liquid form is getting converted to the gaseous form. They have asked us about the correct set of thermodynamic parameters. Now how do we do it? So as you can see, the reaction is in equilibrium, so delta G value will be equal to 0 because the reaction is in equilibrium. Other than that, if you look at the change in entropy, you can see that the water is changing from the liquid to the gaseous phase. This means as the water changes from liquid to the gaseous phase, there is more randomness or disorderness in the system, so the delta S value becomes positive. So the answer is B. Let's go to the third question. The third question is based on statements. All right. So which of the following statements are correct? So the first statement is based on the Clausius-Clapeyron equation, which correlates the enthalpy of vaporization and the partial pressure, and it's correct. The second option is on Troughton's rule, which is nothing but the ratio of the enthalpy of vaporization to the boiling temperature, that is equal to 88 joule per mole per Kelvin, which is also correct. And entropy is a measure of unavailable energy. Unavailable energy is the energy which is unavailable to do useful work. So D, that is all of the above statements are correct. This is the answer. Moving to question number 4. The question number 4 is based on relationship between enthalpy and internal energy. So the question is, we have N2 plus 3H2 giving us 2NH3. This was carried out at constant temperature and pressure. If delta H is the enthalpy change and delta E is the internal energy change, respectively, which of the following expression is correct? So what is the relationship between delta H and delta E? Delta H is equal to delta E plus delta Ng RT. So if you have to calculate delta Ng, that is the change in number of gaseous molecules, so that will be equal to the number of Np minus Nr, that is the number of moles of the gaseous product minus the number of moles of the gaseous reactant, that is equal to 2 minus 3 plus 1, that is 2 minus 4, that is minus 2. So if you calculate delta H comes out to be delta E minus 2RT. So can we just say that delta H value will be less than delta E, right? So C is the correct answer. Now let's talk about the question number 5. Question number 5 is based on Hess's law. So as you can see, there are two expressions. One in which Fe2O3 plus 3CO gives you 2Fe plus 3CO2. Here the delta H value is minus 26.8 kilojoule. And the next equation, this is equation number 1 and this is the equation number 2. The next equation is FeO plus CO gives you Fe plus CO2. Here the delta H value is minus 16.5 kilojoule. From the above two reaction, you have to calculate the value of delta H for the following reaction. So very simply we can do this question. As you can see, look at the reactant side. On the reactant side, you can see Fe2O3 plus CO. So which matches, which tallies with our first expression. So we will not change the first equation. In the second equation, basically if you see the product side, you will see that the FeO and CO2 are on the product side. And FeO is multiplied by 2. So which gives us an indication that we need to reverse the equation number 2. So if we reverse the equation number 2, what are we going to do? So we are going to get this expression. And as you have reversed the expression, the delta H value changes to plus 16.5 kilojoule. So the next thing that you have to see is the FeO is multiplied by 2. So we will just multiply this delta H by 2. So if you multiply the delta H by 2, you're going to get this, right? So you're going to get this. Now what are we going to do? To calculate the value of delta H, we'll just add the content of 1 and 2. So if you just add the content of 1 and 2, you are going to get this expression. So if you add minus 26.8 kilojoule plus 16.5 times 2, you are going to get the answer of 6.2 kilojoule. So the correct answer is B, 6.2 kilojoule. So this question is based on Hess's law. Now the sixth question. In the sixth question, when 2 moles of an ideal gas, as you can see, the number of moles is given to you as 2. The Cp value is given to you as 5 by 2R, is heated from 200 Kelvin to 400 Kelvin. So here T2 value is 400 Kelvin and T1 value is 200 Kelvin. Now the change in entropy is something that you have to calculate. So change in entropy will be given by this relation, NCP ln T2 by T1, right? So what is the value of N? The value of N is 2. What is the value of Cp? The value of Cp is 5 by 2R ln of T2 by T1. So it will be 400 by 200, right? So if you calculate, you are going to get 5R ln 2 as the answer. So as you can see over here, 5R ln 2 is the answer. Now this question is actually based on the thermodynamics again. So the correct answer is 5R ln 2. So let's talk about the question number 7. In the question number 7, 18 gram of ice is converted into water at 0 degrees Celsius and 1 atmosphere. So the relation, so the equation is pretty simple. Ice is converted to liquid water. This is what is happening. The entropy of water solid is given as 35.2 joule per Kelvin per mole, and the entropy of liquid is given as 60 joule per Kelvin per mole. You have to calculate the enthalpy. So for this we can use Gibbs-Helmholtz equation, delta G is equal to delta H minus T delta S, right? So here we have to calculate the value of delta H. Delta G will be equal to 0 since the reaction is at equilibrium. So if you have to calculate delta H, so delta H minus T delta S will be equal to 0. Delta H is equal to T delta S. And if you have to calculate delta H now, T over here is 0 degrees Celsius. So you will just do 0 plus 273, that is 273 Kelvin, right? So delta H will be 273 times delta S. Now how do we calculate delta S? We will be just doing product minus the reactant. So this is how we are going to get 24.8 joule per Kelvin per mole. So if you solve this, you're going to get the answer. So this is the method to calculate the enthalpy using Gibbs-Helmholtz equation. So if you have to calculate, you get the answer as 6770.4 joule per mole. Let's talk about question number 8. Here the reaction, for a reaction, delta H is equal to 35.5 kilojoule per mole, delta S is equal to 83.6 joule per Kelvin per mole is given. The reaction is spontaneous at, so basically as you know, if the reaction has to be spontaneous, then the delta G value will be less than 0, it will be negative. So let's use the Gibbs-Helmholtz equation again. So delta H minus T delta S should be less than 0. If that is the case, then we can just say that delta H will be less than T delta S. We can say that T will be greater than delta H by delta S. So delta H value is 35.5 kilojoule per mole. So we will just be doing 35.5 times 10 to the power of 3 divided by delta S, that is 83.6. So if you calculate this, you are going to get T will be greater than 425 Kelvin. So this is how we solve the question and we get the answer as T greater than 425 Kelvin. Let's talk about question number 9. So in the question number 9, which of the following is not a general characteristic of equilibria involving physical processes? Now this question is based on equilibrium chapter. Now let's look at the options. The first option is equilibrium is possible only in a closed system at a given temperature. So basically the first statement is correct. Here the equilibrium is possible in a closed system. All measurable properties of the system remains constant, that is all measurable properties like temperature, pressure and volume, they are all constant. The third option, that all physical processes stop at equilibrium, is incorrect because physical processes, the equilibrium is dynamic in nature. So both the forward and the backward reaction occur at the same rate. So C is the correct answer. Let's just read the D option as well. The opposing processes occur at same rate and there is dynamic but stable condition, which is correct. So the only incorrect statement is that of C. Let's talk about the question number 10. Now, which of the following statements is incorrect? Now let's see the statements first. The first option says, an equilibrium mixture of ice and water kept in a perfectly insulated flask. So when we are talking about perfectly insulated flask, there is no exchange of mass and energy. So mass of ice and water does not change with time is the correct statement. Next option is the intensity of the red color decreases when oxalic acid is added to a solution containing iron 3 nitrate. So basically, if we have iron 3 plus and if we just add thiocyanate to it, we get a complex that is FeSCN thrice. Now this complex, because of which the complex basically because of this reaction the complex is formed and the complex is red in color. The intensity of the red color decreases when oxalic acid is added. So basically if you add oxalic acid to Fe3 plus gets involved in reaction with oxalic acid. So as the Fe3 plus's concentration decreases, the intensity of the red color will decrease. Now this is also a correct statement. Now on addition of catalyst, the equilibrium constant value is affected, which is not true. Catalyst does not affect the equilibrium constant value, it only affects it alters the energy of activation. So C should be the correct answer. let's just go through D as well. The equilibrium constant for a reaction with negative delta H value decreases as the temperature increases, which is true. When delta H is equal to negative, the reaction is exothermic in nature. For exothermic reactions, if you increase the temperature, the equilibrium constant will decrease. Now let's talk about question number 11. Which of the following will produce a buffer solution when made in equal volumes? Let's see the options. The first option is 0.1 mole per decimeter cube ammonium hydroxide and HCl when they are in equimolar concentration. The C option has 0.1 mole per decimeter cube ammonium hydroxide and 0.05 mole per decimeter cube of HCl. While B and D will have, as you can see in case of B, the concentration of ammonium hydroxide is less, and in case of D, equimolar concentration of sodium acetate and NaOH is given to you. What is the concept behind it? Now, buffer solution is that solution which resists any change in pH on addition of H plus and OH minus. Any weak acid and its salt, weak base and its salt will act as a buffer solution. Now, C is the correct answer. Why? Because we have 0.1 mole per decimeter cube of ammonium hydroxide and we have HCl around 0.05 mole per decimeter cube. Now what is happening over here? The HCl is reacting with ammonium hydroxide and giving ammonium chloride. So basically around 0.05 mole per decimeter cube will react with 0.05 mole per decimeter cube of ammonium hydroxide and give ammonium chloride. This is the salt. And while this reaction takes place, some of the ammonium hydroxide remains unreacted. So now we have the solution of ammonium hydroxide, which is a weak base, and we also have ammonium chloride, which is a salt. So this can act as a buffer solution. Let's talk about question number 12. Here the Ka for acetic acid is 1.5 times 10 to the power of minus 5. Kb is again same value. You have to calculate the pH of the ammonium acetate. So for this we use this formula, pH is equal to 7 plus half pKa minus pKb. Right? Since Ka and Kb are having same value, so this quantity will get cancelled. So pH should be equal to 7. So this should be the answer. So the correct answer is 7. Now let's talk about the next question. Which of the following equilibrium remains unaffected on addition of small amount of neon at constant volume? So here, what are we talking about? So here we are actually talking about small amount of inert gases at small amount of addition, basically addition of inert gas at constant volume. As you know, addition of inert gas at constant volume will not affect any kind of equilibrium, but addition of inert gas at constant pressure will affect the equilibrium. The equilibrium will move in the direction where there are more number of gaseous molecules. So over here, as you can see, since they have mentioned constant volume, so the correct answer has to be D. The equilibrium will remain unaffected in all the three cases. Let me talk about the number 14 question. Now this question is based on redox reaction. Here we'll just show you two ionic equation and we'll try to solve the problem. How many moles of KMnO4 are reacted with 1 mole of ferrous oxalate in acidic medium? Now how do we solve the question? For this we need to use we need to know how to balance the equations, how to balance half equations, how to write the ionic equation. So this is based on two ionic equation. First, MnO4 minus plus 5, since ferrous is there, so Fe2 plus will be converted to Fe3 plus, MnO4 minus will be converted to Mn2 plus. These are the two half equations. If you solve it, you're going to get this expression. This is one expression. This involves ferrous. When we talk about oxalate, So 5C2O4 2 minus, this is oxalate. So these are the two expressions. So if you look into these two expressions, you can easily see that one, if we just look at the moles of KMnO4, 3 moles of KMnO4 basically react with 5 moles of ferrous oxalate. If that is the case, so if we are talking about 1 mole of ferrous oxalate, so 1 mole of ferrous oxalate will react with 3 by 5 moles of KMnO4. So the correct answer should be 3 by 5. So C is the correct answer. Now let's just discuss the question number 15. Here the oxidation number of sulfur in H2S2O8 has been asked. H2S2O8 is peroxo disulfuric acid. So the formula of H2S2O8 is, since it is peroxy disulfuric acid, so there has to be peroxide bond. Okay, so if you have to calculate the oxidation number of sulfur, so it will be 2 times X. How many oxygen are there? 1, 2, 3, 4, 5, 6. 6 in the form of oxide. And there is a peroxide bond. So 2 into minus 1. 2 hydrogens are there. So 2 into 1. So if you solve it, you are going to get 2x is equal to 12. X is equal to 6. So the oxidation number of sulfur in H2S2O8 is 6. So this is the answer. So correct answer is C plus 6. Let's talk about the question number 16. Now, which of the following rules is not correct for oxidation state? Let's just go through the options. The oxidation number of hydrogen is always plus 1. That's not true. In case of hydrides, the oxidation number is minus 1. So A should be the correct answer, but let's just go through B, C, D. In B, all the oxidation numbers in neutral compound is 0. That's true. C, an element in the free or the uncombined state bears oxidation number 0. That's also true. In all its compounds, the oxidation number of fluorine is minus 1. Fluorine is the most electronegative element in the periodic table. That's true. So A is the correct answer. Now let's talk about question number 17. Now here the pH of the buffer containing 2.4 gram of acetic acid and 8.2 gram of sodium acetate in 1 liter of water is. So this question is based on Henderson-Hasselbalch equation, which is pH is equal to pKa plus log of salt by acid. Right? So here the salt is sodium acetate, acid is acetic acid. So let's just first calculate the concentration of the salt. The concentration of salt, as you can see, concentration is number of moles by volume in liters. Now volume they have given 1 liter. So whatever the number of moles is there, that will be the concentration of the salt. So this will be equal to 8.2 divided by 82 because I'm using given mass upon molar masses formula. So this gives us an idea of this gives us the number of moles of 0.1. If you have to calculate the concentration of acid, so this will be again 2.4 divided by 60. That gives you a value of 0.04. pKa value is 4.5. Let's calculate the pH. So pH will be equal to 4.5 plus log of 0.1 divided by 0.04. So if you solve it, you are going to get 4.5 plus log of 2.5. So this should be the correct answer. Okay, so let's try to solve. Let's just see whether this tallies with the answer or not. So B is the correct answer. So the next question is based on solubility product concept. The solubility product of barium hydroxide is X. Then you have to calculate the value of OH minus in saturated solution. So basically barium hydroxide, if you just dissolve barium hydroxide, you're going to get barium 2 plus aqueous plus 2OH minus aqueous. Let's say the solubility of Ba2 plus is S, then OH minus will be 2S. So KSP will be equal to the product of the, basically it is the product of the concentration of the product. So it should be S times 2S, since the stoichiometric coefficient is 2, so it will be raised to the power of 2. So this gives us 4S cube. Now the KSP value is X. So we can just write S is equal to X by 4, 1 by 3. Okay, now if you have to calculate OH minus is 2S, that is 2 times X by 4. The cube root of X by 4. So the correct answer over here is B. Let's take the 19th question. What will be the solubility product of AX cube? So if you dissolve AX cube in water, you're going to get A3 plus aqueous plus 3X minus aqueous. Right? If you have to calculate the KSP, again, what will be the KSP? It will be, let's just say the solubility of A3 plus is S and the solubility of X minus is 3S. Right? So it is the product of the concentration of the product. So S raised to the power of its stoichiometric coefficient. So this will give you an answer of 27 S to the power of 4. Right? So this is the answer. So the correct answer is D. Let's take the 20th question. The amount of H2O2 present in 1 liter of 1.5 normal H2O2 solution is. So we have to calculate the strength of H2O2 solution. This is nothing but normality into the equivalent weight of H2O2. Normality is 1.5. Equivalent weight of H2O2 will be 34 divided by 2, that is 17. So if you calculate, you are going to get 25.5 gram per liter. Now since they have asked in 1 liter the amount of H2O2, so the answer is 25.5 gram, right? So D is the correct answer. Let's take question number 21. Now this concept is based on S block elements. Correct order of thermal stability of hydrides of alkali metals. So as you know, hydride, if we talk about the stability of hydride, down the group the stability of hydride decreases because the size compatibility is less, the lattice energy will be less of the hydride so formed. So the correct answer has to be A, where lithium hydride is the most stable and rubidium hydride is the least stable. Let's take question number 22. Among the following hydrides, choose the one which is electron precise hydride. Now covalent hydrides are classified into 3 types of hydrides: electron precise, electron rich and electron deficient. Electron rich are those which have lone pair of electrons. Electron deficient will have incomplete octet. So NH3 and H2O are electron rich. B2H6 over here in the B part is electron deficient. So D, SiH4 is the answer. Silicon, silicon hydride is an electron precise hydride where sufficient number of electrons are present. Now let's take the question number 23. The question is very, very simple. It's from hydrogen chapter. What causes permanent hardness of water? So the options are A, chlorides and sulfates of magnesium and calcium, bicarbonates of magnesium and calcium, carbonates of magnesium and calcium, and nitrates of sodium and potassium. So A, chlorides and sulfates of magnesium and calcium will cause permanent hardness. They will not be removed by boiling. So A is the answer. Now question number 24. Out of the following alkaline earth metal, choose which one does not impart color to the flame. So if you get a question like this, you need to look for the smallest smallest among the alkaline earth metals. So over here the smallest ion is magnesium. So magnesium is smallest in size. It will bind the electron very strongly, so it will require very, very high excitation energy. So this magnesium will not impart any color to the flame. So C is the correct answer. Now taking the question number 25, the average kinetic energy in joules of the molecules in 10 gram of oxygen. So here they have given 10 gram of oxygen at 27 degrees Celsius is how much? So this question is based on kinetic theory of gas, states of matter. So kinetic energy will be given by 3 by 2 nRT, where 3 by 2, n is the number of moles, given mass 10 upon the molar mass 32, R gas constant 8.314, and the temperature is 27, so just add 273 to it. So this will give you the correct answer of kinetic energy. So if you solve it, you are going to get 1169.2 joules as an answer. Now let's talk about the next question. The next question is a graph-based question. Now this is also based on the gas laws. Now for the given mole of gas, the correct order of pressure for the following isobars, this is the question. The correct answer is B. Here P1 is greater than, if you look at the graph, P1 is greater than P2 is greater than P3. This graph is based on Charles' law, where volume is directly proportional to temperature at constant pressure. So as you can see over here from the graph, we can very well say that P1 is greater than P2 is greater than P3. Now let's talk about question number 27. This is a very simple question based on redox reaction. The equivalent weight of potassium permanganate in neutral medium is, so when we are talking about neutral medium, MnO4 minus gets converted to MnO2. Let's look at the oxidation number. Mn has the oxidation number of +7 in MnO4 minus. In MnO2, the oxidation number is +4. As you can see over here, 3 electrons is basically the number of electrons required will be 3. So if you have to calculate the equivalent weight, so equivalent weight will be the molecular weight, that is M divided by 3. So D is the correct answer. Now the next question is based on, this is in GOC, this is based on general organic chemistry. The major product A in the above reaction has been asked. Let's try to solve it. As you can see over here, there is an addition. They have given you CH2OH. So there is an addition of H plus taking place. So what will H plus do? H plus will attack, basically H plus is electron deficient. So basically H plus will attack the electron rich oxygen and you're going to get OH2 plus. Now we'll remove the OH2 and this will generate our carbocation. Now as you can see, this is a 5-membered carbocation with a CH2 plus. Now here a ring expansion will take place in order to give the stable carbocation. So this particular bond will migrate. Okay, so how do we understand where will the plus sign go? Okay, so we'll try to draw the structure. So as you can see, 1 and 6 will form a bond. So I'll draw a 6-membered ring, 1, 2, 3, 4, 5, 6. Now as you can see, the bond has gone from 6 to 1. So the plus sign appears over here. Now I'll just put from here. As you can see from here, what will I do? I will just heat this. Now if you heat this, what will happen? There will be a loss of H plus and we are going to get B, cyclohexene. So this is how we get the answer. Now let me take question number 29. the correct increasing order of dehydration of the following alcohol with concentrated H2SO4. The concept is again the same since we are talking about dehydration. In the previous case also we were talking about dehydration. We tried to produce a stable carbocation. So again the reactivity of alcohol will be totally dependent upon the stability of the carbocation. So as you can see over here, we have A, B, C. As you can see, if the OH, basically what is happening over here, there's a simple trick to do it. So over here, wherever there is an OH, okay, wherever there is an OH, as you have seen in the previous case, wherever there is an OH, I produced water and I just removed the water. So wherever there was an OH, I just put a plus over there and that will give you the stable carbocation. So wherever there is an OH, just put plus over here. Just remove the OH and just put a plus. Okay, now let's judge it. So if you look at the D carbocation, so basically if you form alkene out of it, a stable, basically an aromatic product will be formed. So D is the most, basically D, if we talk about the increasing order, in D the reaction will take place smoothly. The dehydration of alcohol will take place very smoothly, very easily. Then comes A. In the A case, you can see that if you remove the OH, you get a secondary carbocation which is stabilized by conjugation. So this gets priority. So D, the reaction will be easier. In A, next the reaction will be easier. Then comes C, because a tertiary carbocation will be formed, though there is no stabilization of conjugation. And then comes B, if you remove the plus, there will be a secondary carbocation form. So basically, depending upon the stability of carbocation, we can say that this is the correct order. So depending upon the stability of carbocation, we can just say this is the correct order, where D is the most stable, then we have A, then C, then B. Now the next question is super simple. Which of the following is meta-directing for aromatic substitution reaction? So the correct answer is A, right? It's a benzene nitrile. So this is the only meta-directing out of the lot. The next question is, which of the following is the most stable carbanion? So carbanion will be stabilized by electronegative group. Any group which will withdraw electron will stabilize the carbanion. So in case of D option, as you can see, D is the only option where 2 electron withdrawing group, that is 2NO2 is present. Since 2NO2 is present, so I can just say that D will have the most stable carbanion, right? Now let me take question number 32. As you can see over here, the reaction, here propene is treated with HCl in the presence of peroxide and you get 2-chloropropane. They have asked the reaction intermediate in the above reaction. So there are carbanion, free radical, carbocation or carbene is the option. So C, carbocation is the correct answer. This is because HCl in the presence of peroxide is there. The first thing, if you see peroxide, the first thing that comes in your mind is it should be free radical addition. But free radical addition takes place only if HBr and peroxide's combination is there. When HCl and peroxide is there, peroxide will not generate a free radical. Instead, HCl will proceed in the route where carbocation formation takes place. So, C is the correct answer. So the next question is, which of the following has the highest boiling point? The concept is, boiling point is directly related to the number of carbon atoms and is inversely related to branching. So as you can see over here, 5 carbon atoms, A has 5 carbon atoms, so 5 carbon atoms is in the linear chain will have the highest boiling point as compared to D. B and C has branching, so D has only 4 carbons, so the boiling point will be highest for the one which has 5 carbon atoms, that is A. Let me take the question number 34. Now in the given Newman's projection formula of 2,2-dimethylbutane, A and B respectively can be. Okay, so let me just draw the structure first. This is 2,2-dimethylbutane. So if we draw the sawhorse projection, let me just draw it in this manner only. Okay, so let me take this first. So CH3 comes over here, there is H, H, and over here all of them are CH3. So this is our A, this is our B. So if you look into the Newman's projection, D is the correct answer, where A, as you can see, A will in the sawhorse projection also we can see A and B are CH3. So CH3, CH3, D is the correct answer. Let's talk about the next question. Let's take the next question. Most stable carbocation is. Over here, as you can see, there are 4 types of carbocation. There is a tertiary carbocation in the B case. In the C case, there is a benzyl carbocation. In the D case, there is a secondary carbocation. Obviously, D cannot be the answer. Okay, it can be either A, B, and C. But as you can see in the A case, there is a cyclopropyl carbocation. What does a cyclopropyl carbocation do? It is considered as one of the most stable carbocation because it shows dancing resonance. Okay, so basically A, the one which has 3 cyclopropyl group attached to it, this is the most stable carbocation because it has cyclopropyl cation attached to it. Right? So this has more stability over benzyl carbocation or tertiary carbocation. Let's take question number 36. Pyridine has less basic character than triethylamine. Okay, so what is pyridine? This is pyridine. What is triethylamine? This is triethylamine. As you can see, the nitrogen in pyridine is sp2 hybridized, and this is sp3 hybridized. Greater the percentage S character, lesser will be the basicity. So sp3 hybridized triethylmethane should be more basic because it has less S character. So this is the logic. Let's try to see whether it tallies with the options or not. Pyridine has aromatic character. Pyridine is a cyclic system. The C option. In pyridine, lone pair of nitrogen is delocalized. D, nitrogen in pyridine is sp2 hybridized. As mentioned, D is the correct answer. Let's take question number 37. Which of the following is correct with respect to -I effect of the substituent? So as you can see, the logic behind it is very simple. As the electronegativity increases, the basically -I effect is directly related to electronegative. So as the electronegativity of fluorine is more than OR, then as you know, fluorine is the most electronegative element. So as the electronegativity increases, the -I effect also increases. So the correct order has to be this. First fluorine, next we have OR group, then we have NH2 group. Let's take question number 38. Now here, what do you have to do? You have to arrange the following in decreasing order of enthalpy of hydrogenation. The logic is enthalpy of hydrogenation is inversely related to the solubility, it is inversely related to the stability of alkene. Greater the stability of alkene, lesser heat of hydrogenation, lesser heat will be evolved. So the enthalpy of hydrogenation will be less. Keeping into consideration this concept, first, as you can see in the first case, you can see that it's an aromatic compound. So obviously the enthalpy of hydrogenation will be least because it will be the most stable form. Next we have, as you can see, next we have 2 and then we have 3 where all the bonds are in exo. Okay, so the logic is enthalpy of hydrogenation is inversely related to the stability of alkene. So 3, that is the third compound. The third compound is having the maximum enthalpy of hydrogenation, followed by 2, and then we have 1, which is the most stable alkene. It will have the least enthalpy of hydrogenation. Now let me take question number 39. So over here, Amongst the given compound, choose the one in which all the atoms are in possible conformation. So as you can say, they are in one plane in all possible conformations. So basically where in A, C and D, the atoms will not be in one plane. There is a reason behind it. As you can see, we have 1,3-butadiene. There is a CC bond. So if there is a CC bond, there can be millions of conformation. Now, in the case of B, you can see a triple bond. There's a C triple bond. So as you can see, C triple bond imparts, basically it gives a linear structure. So if there is a linear structure, then the conformation possible, basically all the possible conformation will be in one plane. Okay. Now that is the logic. If sp hybridization, if you are talking about sp hybridization or alkyne, there can be a confusion why D is not the answer. This is because in D, the C is sp hybridization, but the CH2, these two CH2s are sp2 hybridization. So here they will be perpendicular to the plane. So the correct answer is B. Okay. Here, because of the triple bond, because of the linearity in the structure, here the atoms will be in one plane. Let's take question number 40. A compound which releases carbon dioxide most readily. Concept behind it, very simple. We are talking about the thermal stability of metal carbonates. The metal carbonate which is least stable will give carbon dioxide most readily. So what are the options? Option number A is magnesium carbonate, B is sodium carbonate, C is barium carbonate and D is potassium carbonate. So sodium and potassium carbonate, they belong to alkali carbonates. So alkali metal carbonates are much more stable as compared to alkaline earth metal carbonates, because of which we reject B and D. Now we have A and C. Carbonate, the stability of carbonate increases down the group. Again, logic is very simple. As carbonate is big in size, the size compatibility will be with a bigger cation, right? So down the group, the lattice energy will be more. So barium carbonate will be thermally more stable. That is why magnesium carbonate will release carbon dioxide easily, because it is least stable. Now let's take question number 41. Identify the compound which is not employed in the process of removal of, basically identify the method which is not employed in the process of removal of permanent hardness. So we have Calgon's method, ion exchange method, Clark's method and synthetic resins method. Over here, Clark's method is the correct answer. This removes temporary hardness. Let me take question number 42. Alkaline earth metal which do not combine with hydrogen to form their hydrides. Okay, again the logic is pretty simple. Let's see the options first. Barium, beryllium, calcium and strontium is there. Generally the electronegativity difference of beryllium and hydrogen are pretty small, because of which beryllium does not combine with hydrogen to form an ionic hydride. Okay, that is why B, beryllium is the answer. Let me take the next question. The next question is based on van der Waals equation of state. So the constant B in van der Waals equation for 1 mole of real gas signifies what? So basically B is the correction in volume term. So since it's the correction in volume term, so it actually denotes the effective volume of the individual gas molecules. So the correct answer is B, volume of the gas molecules. Okay. Now let me take question number 44. Now which of the following statements is incorrect? Let's see the statements. First statement is, at high altitude, liquid boils at higher temperature in comparison to that of sea level. This is incorrect. Why? Because at higher altitude, the atmospheric pressure will be low. So as a result of which, liquid will boil at a lower temperature in comparison to that of sea level. So this is an incorrect statement. So let's just go through B, C and D to see whether they are correct or not. So in B, boiling does not occur when liquid is heated in a closed container, which is true. Next, at critical temperature, the density of liquid and vapor becomes same. That is also true. And surface tension is inversely related to temperature. Surface tension of liquid decreases as the temperature is raised. That is also true. So A is the incorrect statement. Now let me take question number 45. Lecine's extract is boiled with concentrated HNO3 before we use it for testing of halogen. What is the purpose of boiling it with concentrated HNO3? Lecine's extract is also called sodium fusion test. This test is also known as sodium fusion test. Here, what do we do? Here, the lecine's extract is boiled with HNO3 to remove any kind of sulfur or nitrogen present in the molecule. So, sulfur gets converted to Na2S. So, nitrogen gets converted to NaCN. So, whatever Na2S and NaCN is present in the solution, this will get decomposed if you boil the extract with concentrated HNO3. So, C is the correct answer. Now, let me take the next question, that is question number 46. Which of the following condition will shift the reaction in forward direction? Let's go through the option. As you can see, the reaction is an exothermic reaction. Increase of temperature will not move the reaction in the forward direction. It will move the reaction in the backward direction. B option is decrease of pressure. So if you decrease the pressure, the reaction moves in that direction where there are more number of moles, that is backward direction. Next, addition of inert gas at constant pressure. As we have just studied, inert gas at constant pressure will shift the equilibrium in backward side where there are more number of moles here. So what will happen if you add HCl gas? So if you add HCl gas, the NH3 will get converted to NH4Cl, okay? As a result of which, what happens? The NH3's concentration decreases. So if the NH3's concentration decreases, then the reaction will move in the forward direction according to the Le Chatelier's principle, right? So let us take the next question, 47. The next question is based on the concept of bond energy. For the reaction, we have hydrazine plus H2 giving us 2NH3. Calculate the bond, basically you have to calculate delta H naught for NH3. Now, given bond energies are Nn is X, then hydrogen is Y, and bond energy of NH is Z. So how do we calculate it? So basically, bond energy is nothing but the summation of bond broken minus summation of bonds formed. So this is basically summation of reactants, the bonds in the reactant minus the bonds in the product. So if you look at the reaction again, hydrazine has been taken, treated with H2, giving us 2NH3. Right? So in the reactant side, there is one Nn bond, there are 4 NH bond, and there is one HH bond. In the product side, there is 2 into 3 NH bond. So what will be the answer? So the bond energy or the bond formation of NH3 will be given by, so in the product side we have N, so for the N the value is given X. Now how many NH are there? There are 4 NH bonds, so the value is Z, so we'll just put 4Z. For HH the value is Y, so we'll add Y and then we'll subtract this by 6 times NH value. So 6 times NH value, NH is Z. So if you solve this, you're going to get X minus 2Z plus Y. So the correct answer is A, X minus 2Z plus Y. So this is how you calculate bond energy as well. Let me take question number 48. Now this is based on the stability of carbocation. So over here, you can see there are 4 types of carbocation. You have to arrange it in increasing order of stability. The lowest should be written first. So as you can see over here, it is very simple to find out the lowest one. What will be the lowest one? The one which has a electron withdrawing group, that is this one. So the one which has an electron withdrawing group will be the lowest one. So D will be the lowest. Okay, so if D is the lowest, next what will we have? We'll have meta, as you can see, the OCH3 group in this case is present in the meta position. So in the meta position, there is no plus R effect, only inductive effect keeps into play. So as a result of which, after D, A should come, then we have benzyl carbocation. And in the next case, we have basically the one which is more stable is that benzyl carbocation where the OCH3 is present in the para position, right? Where the OCH3 is present in the para position, that is the most stable carbocation. So B is the most stable carbocation, followed by C, then we have A, and then we have D. So if you want to do these questions very easily, first find the lowest stable carbocation and then find the highest stable carbocation and then look for the correct option. That will save a lot of time. Now let me take the 49th question. So the state of hybridization of C1, C3, C4, C5, C6 of the hydrocarbon we have to find, which is pretty simple to do. Now as you can see, they have asked about C1. C1 has sp hybridization. We have C3, sp3 hybridization. Next we have C4, that is sp2 hybridization, and then we have C6. This is sp3 hybridization. So the correct answer is sp, sp3, sp2 and sp3. So this is how we solve it. Now let me take the 58th question. What is the correct order of the stability of the carbon ion? So the stability of the carbon ion will depend upon the percentage of S character. The one which has the highest percentage of S character will be the most stable carbon ion. So out of the lot, we can very well make out that A is most stable, right? A should be the most stable carbon ion, and the least stable carbon ion has to be the one which is R3C CH2 minus. So this should be the least stable because it has sp3 hybridization. So the correct answer over here is A comes first, followed by C, which has sp2 hybridization. Then we have B, and then we have D. So this is how the stability order of carbon ion is seen, right? So the concept over here is pretty simple. Carbon ion, the stability of carbon ion will be directly related to the percentage S character. So these were the questions from the test, AIRTS test minor test 2. I hope this was helpful to you. Let's meet again while discussing the questions from AIRTS minor test 3, which will be conducted on the 26th December. Thank you so much.
Good afternoon, everyone. Welcome to the Physics Minor Test 2 discussion, which I'm sure you're familiar, and I hope you have given the test also, Minor Test 2, that was conducted this weekend, day before yesterday only, and some of you might have given yesterday. So now what we are going to do is we are going to discuss all the physics question, all right, very quickly, and I'm sure you're familiar with the syllabus. So what was the syllabus? The second part of the 11th class. All right, basically in the first minor test we had a question from mechanics, right, from unit and dimension to rotational, and in this test we had questions from mechanical properties of solids all the way to waves and oscillation, including thermodynamics, of course. All right, guys, so let's begin for physics. And those of you who are watching it live, okay, maybe some of you may not have given the test, but still the sessions are going to be definitely helpful for you also, because what we are going to do is we are solving the questions. All right, so even if you have not given the test, you can try solving the questions during the live class, although we'll go a little quickly, so you won't have the time to actually solve it, but you can look at the solution and you can try to understand the concepts that are involved in those questions. All right, guys, let's begin. Okay, first question. So this question is basically about what it is asking. So it is asking g is acceleration, all that, change in potential energy. All right, that is what it is being asked here. So we need to basically apply the formula of potential energy. So it is asking for change. So how do we find change? That is the final potential energy minus the initial potential energy. All right, and what is happening here is that m mass, earlier it was at the surface, and now it is a very large height, and the height is equal to the radius of that. So if we draw the diagram, so initially the mass was at the surface only, but finally, where is the mass? The mass is raised to a very large height, and that height is basically equal to the radius of the earth, right, finally. So this is basically same as the radius of the earth. Now some of you might have done this mistake that you have written change in potential energy, that is equals to mgh, where you say that the height is equals to the radius here, so mgr, which means C, that should be the right answer, right? But that would be incorrect, because mgh, this formula, is not valid when the height is very large. All right, why is that? Because actually the small g is does not remain constant. You might be knowing that it varies with the height. So if at a large height, the small g will not remain constant, so that is why this formula will not be valid anymore. So that is why we cannot apply mgh for large height. It is applicable only for small height. So for large height, what is the formula we'll use? So the formula is very simple, guys, that you must remember. The potential energy of a mass particle, small m mass, at a distance r, let's say small r, the particle is here, let's say, okay, at a distance of small r, so its potential energy is given by minus g, mass of the earth, capital M, mass of the body, divided by r, small r, that is the distance from the center. All right, so that is the formula, very, very important one, and that is what we are going to use in this question, and that will give us the answer very quickly. So we'll put the values. Change it is asking, so let me find the final value, potential energy final. What is that? GMM. Okay, distance from the center. Finally, what is the distance? The distance is r plus r, 2r. All right, so that is the final value. Let me write the initial one also. Potential energy initial, that is minus g, capital M, mass of the earth, small m, mass of the body, divided by distance. Initially, what was the distance, guys? When the particle was at the surface on the earth, what is the distance between the earth and the particle? So the distance is same as the radius, right? When we are at the surface of the earth, how far we are from the center of the earth? Radius of the earth, so that will be r. All right, so now we want to find change in potential energy, that is final minus initial. So final is minus g m small m by 2r minus initial, that is initial, so minus will become plus actually later, so that is why we are getting a positive answer. So this will become plus, so plus gmm by r minus gmm by 2r. So what will that be? That will be gmm by 2r. Is that my final answer? No, because the options are not given in this way. The options are given in terms of small g. So very simple. We can write capital M and capital G in terms of small g, and that will be we can write capital GM is gr square, small g r square into m divided by 2r. So r and r will get cancelled and we'll get g r m divided by 2, right? This r and this power will get cancelled. Okay, some of you might be confused, sir, how did you write this one from capital GM? So guys, that is the formula of small g, acceleration due to gravity. What is the formula? You must be remembering this one. This is important. Small g equals to gm by r square. This is the formula of acceleration due to gravity. So yeah, that is what I have written here. Capital G, capital M is small g r square. So small capital G, capital M is equals to small g r square. So now then r and this power get cancelled. So we have g r m divided by 2. That is the answer, which will be option A. Rest of this formula very, very important. This one also very, very important. Rest was just calculation that anyone can do. Alright guys, let's continue. In case you have any doubt in between also, you can put it in the chat, guys. I'll be keep looking at the chat also. So okay, next one, escape velocity. This was kind of a trick question, I would say. So what is the question? They are asking escape velocity and at what angle? At 45-degree angle. So they are saying, okay, we launched a object at a 45-degree angle from the vertical, or which is same as from the horizontal. So what is the escape velocity of this? Like, what is the minimum velocity we must give so that it will escape into the space, outer space? So that is escape velocity. So good thing, guys, escape velocity does not depend on the angle. It comes from conservation of energy. If you remember the derivation of escape velocity, the formula also, if you look at the formula, there's no mention of the angle. So it does not depend on angle, basically. That is why answer is D. So escape velocity at the surface for any angle, even if we throw it horizontally also, like the parallel to the surface, even then escape velocity remains same, 11.2 meter per second. So that is why answer is option D. Alright guys, let's continue. Orbital velocity of a satellite is V. What is the orbital velocity at altitude? Half the radius. So this one again, guys, formula based. We need to remember the formula of orbital speed or orbital velocity. Like when the satellite is revolving around Earth, what is the orbital velocity of that satellite? Very, very important formula. I'll write it for you here, and that is V equals to root G M, capital G, capital M is mass of the Earth divided by the R. R is the radius of the orbit. This formula is very, very important. Okay, if you remember this one, then you're done. This question you can do easily. So initially at the surface, if I write, let's say at the surface, so at the surface, what is the distance? Again, guys, that is equal to the radius, right? At the surface, when object is there, its distance, this small r is from the center of the Earth. So at the surface, this is equal to the radius of the Earth. Okay, what about at some height? So let's say V dash, that is what they are asking. So that is root GM, okay, divided by the distance. Now what is the distance? Satellite orbiting at altitude half the radius. So altitude is half the radius. So this is the Earth, this is the radius. What is the altitude? That is r by 2. So altitude, which is height, so that is r by 2. So what is the total distance from the center? That is r plus r by 2, 3r by 2. That is what we are going to put here, 3r by 2. Now we are done. 2 by 3 is the different thing. Root gm by r is common. So root gm by r is basically same as v. So I can write this thing as 2 by 3 into v, right? So that is my, this is the equals to sign. So yeah. That is my answer. So root 2 by 3 into v, which option is that? So that is option C. It's very simple, just based on this one, very important, orbital velocity of a satellite. Alright guys, let's continue. Radius is given for Earth, Mars also is given, mass of Earth is given 10 times, weight of an object is given. What is the weight on Mars? Interesting question. So guys, it requires two formula here. One is weight. Weight, of course, we know mg, right? This is very important one. I hope you remember this one. Weight is mg. So weight is m. Mass does not change, right? Even if you are on Earth, Mars, Jupiter, Andromeda galaxy, it doesn't matter. So mass doesn't change. What changes is the small g, that is acceleration due to gravity. On Earth, it is different. On Moon, Mars, it will be different. So the challenge here is to find the acceleration due to gravity, small g, and it is also very simple if you remember the formula of small g, and that is capital G, capital M by r square. So we'll find it for Mars. So let's try to find it for Mars. Let's say G Mars, M is for Mars. Okay, capital G. Okay, mass. What is the mass of Mars? Mass of the Earth is 10 times. So what is the mass of the Mars? That is mass of Earth divided by 10, right? If this is for Earth, let's say, now I'm trying to find it for Mars. So mass of the Mars is 1 by 10 times of Earth. And what about the radius? Radius and that of Mars, so radius is half, right? It is given radius is half for Mars, so r by 2, it will become r by 2 and square of that. That is my value of acceleration due to gravity on the surface of Mars, right? That is the formula. So now we just need to simplify this one, gm by 10, this will become r square, 2 square, it will become 4, go into numerators, 4 by 10 into gm by r square. So what is that? 4 by 10 into gm by r square. What is gm by r square? That is same as small g only. So this is 4g by 10. That is the value, 4g by 10. So if small g, this is on Earth, let's say. Now let me write it on Mars. That is mass of the body, which remains same, into g on Mars. So this is changing. So what is that equals to? This is equals to mass of the body into gm. What is gm? That is 4g by 10. 4g by 10. So this is 4 mg by 10. 4 mg by 10. 4, what is mg? Mg is the weight on the Earth, that is 200. So this is basically equals to 4 by 10 into the weight, which is 200. So that is my answer, which will be 80 if you solve this. Alright, so yeah, then simple. So basically two formula here, this one and this one. Both are very important, and yeah, rest is just calculation, guys. So the goal here of this, what I'm trying to do here, this discussion minor test 2, I'm not telling you how to calculate that, that is like simple. This is what 8th, 9th class mathematics. That is not what I'm telling you. What I'm telling you is what are the formulas that we need to apply, and that is what we want to learn, right? Because there are very few formulas actually. If you think in gravitation hall, in this chapter there are less than 10 formula. If you remember that and you know how to apply them, that's it. You're done with the concepts. So yeah, guys, let's continue with the same theme. We'll look at the formulas very quickly, and then basically we'll apply it in the question. So distance is given ratio of time period. So guys, what is the formula here? So this is from Kepler's third law. That is T square is proportional to R cube. Or I can also write this thing as T is proportional to radius power 3 by 2. Alright, so now we can see very easily, this is 10 power 12, this is 10 power 13. What is the ratio? Ratio is 10. This is 10 times, right? 10 times. So what about the time? Time will be 10 power 3 by 2 times. 10 power 3 by 2, that is 10 root 10 times. So time of this two planet, the first planet which is at 10 raised power 13 distance, meter distance, the time of that will be 10 root 10 times. So that is the answer, 10 root 10, very simple, right? So the question is simple only if you remember the formula. Okay, this one is a little bit different. Let's see this one, two particles equal mass revolving around a circle. So there are two particles, okay, equal mass. Now they are revolving in a circle. Why? Because of gravitational force. Alright, so kind of weird to imagine that there are two particles, they are revolving in a circle. Why? Because of their mutual gravitational force. There is nothing at the center for they are revolving around. They are revolving like around the center, but because of each other. So guys, this is basically they are undergoing circular motion, and we know that circular motion is possible only because of centripetal force. So there must be a force at the center, towards the center, and that is the centripetal force. And from where is that force coming? That force is basically coming from the gravitational pull of the two masses. The gravitational force that is acting as the centripetal force. So that is what we are going to do. What we'll say is that this is the circle. Okay, the two particles must be at the two ends of a diameter, because only then both of them will experience force towards the center, right? Because they are attracting each other. So they are attracting, and the force is also towards the center. So it means they must be at the two ends of a diameter. So guys, two ends of a diameter. Now basically we'll equate gravitational force with the centripetal force. I hope you remember the formula of centripetal force, very, very important. So first I'll write the formula of centripetal force. What is that? That is mv square by r. Very, very important. Okay, and that is equals to gravitational force, that is g, m1, m2, both are m, so m square divided by the distance. Now what is the distance? Distance is diameter, so that is 2r. So 2r square. That is the equation here. Now you can solve it very easily. You'll get the velocity and you'll get the option C. 1 by 2, why, how 1 by 2? Because as you can see, 2 square is there, and when you take finally we'll take under root, so this will become 1 by 2. Alright guys, so that was the 6th question. Let's go to 7th one. Height R, potential energy. So again guys, simple formula base. What is the formula? Minus G capital M small m by R. Put the formula, get the answer directly. That is why it is in negative, because formula is negative. There is only one option in negative, so that must be the answer, right? That also you can see if you know the formula. Okay, this one. Okay, this question and 9th question also you'll see similar. So guys, what we have to do in such question? If you look at the question, there is a cylindrical metal rod shaped into a ring, small gap is there. Okay, what we are doing is we are heating it. Alright, we are providing heat to it. So what will happen? We know that heat, due to heat, things expand, right? In the that is what we have studied. When we heat something, it expands, whether it is a metal or gases also, right? So this must expand. But if it expands, what about this gap here? What about this X? Will it also expand, or will it contract because that metal is like expanding, so gap must decrease, right? That is what intuitively we might think, that the gap should decrease because the metal is expanding. But guys, the answer is not that, because if you think more logically, if everything is expanding, that the gap itself would also expand. It's like if you think the universe is expanding, then the gap between the atoms should also expand, right? It's not that only the atom is expanding, but the gap is also expanding. So that is what is kind of happening here between the two molecules of this ring, metal ring, between the two atoms. The gap is also increasing. So all the gaps are increasing. So that is why this X gap will also increase. So if I draw the final diagram of this, it will become kind of like this. This ring will become like this. So not just the radius R, but also the thickness of this will also increase. It will become thicker due to expansion, and the distance, the gap distance, that will also increase. So that is what happens. Everything increases, the gaps also increase when we heat. That is the answer to this question, all increase, and that is also the answer to next question. If you see guys, here this part is cut, we heat the sheet, metal sheet, but the gaps also will increase. So that is why D1 and D2 both will increase. That is the answer. So remember that in such, these are trick question, I would say, and in these questions the gaps would also increase. That is what you need to remember. Alright guys, let's continue. 50 gram copper. This is guys, what is saying, copper is there, heated, temperature changes delta 10 degrees Celsius, same heat is given to water. What is the change in that? So guys, very simple formula for heat, and that is Q equals to MS delta T, right? We'll apply this one. Same heat is given, so M water, S water, delta T is equals to this heat is given to copper, so M copper, let's say MCU, S Cu, delta T Cu, right? We know everything, we'll put the values, we'll get the answer. Specific heat of copper is given, okay, and all the values are given, so we'll apply the values and we'll get the answer. It is asking rise in temperature for copper, so this is what delta T is asking. Specific heat of water, guys, I am sure you remember this one. What is that? So for copper it is given, but what is the value for water? So water, it is 4200, right? This I hope you remember, joule per kg per degree Celsius. Specific heat for water. Alright, masses are also given, everything is given. Alright guys, so yeah, put the values, we'll get the answer very quickly. Okay, next one, two liquids, 32 and 24 equal masses, they are mixed, and final temperature is 28. What is the ratio of specific heat? So again guys, same formula. This is basically what we are doing is calorimetry, and what we do in such question is we say heat lost is equals to the heat gained. So we find heat lost and we equate it to heat gained. So what is heat lost, guys? So heat lost, MS delta T, formula is same only. So who is losing the heat? A is losing the heat because its temperature is decreasing. So MA SA delta T, that is the heat gained. Who is gaining the heat? B. So MB SB delta T B, delta T B. So yeah, just put the value. So M masses are equal, so masses get cancelled. Specific heat A delta T for A. What is the delta T for A? Initially it is at 32, finally it is at 28. So delta T is 4. Specific heat of B, delta T for B, what is the change in B? It is at 24, now it is at 28. So change is also 4. So we have to look at the magnitude only, because we are already saying heat lost and heat gained, so we have incorporated the sign automatically. So in such question, guys, calorimetry, when we are equating heat gain and heat loss, you can just take the magnitude. All right, so because we are equating heat lost, so already it is given that heat lost should be negative, but that is included, so you don't need to worry about the sign in this one. That is a good thing. So just find delta T, and delta T, although it is final minus initial, so someone can say, sir, for A it should be negative because temperature is decreasing, but yeah, but here we are looking at the magnitude only, so that is why we have taken 4. So now as you can see, 4 cancel, both are equal, ratio is 1. That is the answer. Alright guys, let's look at this one. Looks complicated, but it is simple, actually very simple. 3 liquid masses are given, specific heat are given, temperatures are given, now they are mixed. What is the temperature? So guys, basically what is again heat exchange is happening, so we can conserve the energy again. These are calorimetry, basically based on conservation of energy. So what we are saying, total initial energy equals to total final energy. So total initial energy, guys, if I talk about, so I can say M1, C1, T1, like Ms, although it is delta T, but if we take T1, let's say initial was 0, so I want to find the total heat of this. What is the total heat contained in M1? So that is M into specific capacity into the temperature. So that is the initial heat in this, plus what is the initial heat in other, C2 T2 plus M3 C3. This is the total heat of the system initially, and that is equals to the final heat, and that is M1 C1 T plus M2 C2. Final temperature becomes same, plus M3 C3 T. So T can take common. So T, what will that be? M1 C1 T1 plus M2 C2 T2 plus M3 C3 T3 divided by M1 C1 plus M2 C2 plus M3 C3. That is option B. Looks complicated, but simple based on conservation of energy. Alright, so yeah, concepts are simple only. It's all about applying them. Okay, this one, heat flux. It is talking about heat flux, joule per second, rate of flow of heat conduction, guys. This is basically, it is talking about heat conduction. What is the formula for conduction? So that is, it is kind of similar to V equals to IR, right? We studied in current electricity. There we study the flow of electric charge. Here it is heat, but formula is kind of similar if you see. So delta V, instead of delta V, we have delta theta, which is change in temperature, which is what the question is asking, equals to I. Instead of I current, we have heat current, which is basically rate of flow of heat, which is, let's say, H, which is heat flux. V equals to IR. So what is R? Resistance rho L by A. So resistivity L by A, but in this chapter we don't use resistivity, we use conductivity, which is inverse of resistivity. So L by A, K is conductivity, of course. So everything is given. Alright, we just need to find delta theta. Everything is given. What? 400. L, what is L? 10 centimeters, so 0.1. Area, what is area? 100 centimeter square converted into meter square. So that will be 10 to the power minus 2 into K, which is 400. So if you solve that, you will get the answer. This is 4000. The H is 4000. So yeah, solve this, you will get the answer, which is C. So yeah, guys, again simple. This formula is very important. So this is the, for heat conduction, this is like the most important formula. Alright guys, let's continue. Black body emit maximum energy at wavelength 14. Temperature is raised, the maximum wavelength, maximum energy. So what is this? What is happening? Maximum wavelength, what are they talking about? So guys, this is basically black body radiation. What we are saying is that if we increase the temperature of black body, the energy it emits, of course it will emit more energy, but if we talk about the particular wavelength at which the energy emitted is maximum, so that wavelength will also decrease. So if basically what I'm saying is, if we are increasing the temperature of a metal body, the wavelength at which it is emitting maximum energy, that will decrease. And what is the relation? So there is very important law, and that is lambda T is equals to constant. What is this called? If anyone remember, this is called Wien's displacement law. Wien's displacement, Wien said that if we increase the temperature, the wavelength gets displaced in the graph, so black body radiation graph. So that is what we need to apply here, the lambda t constants. So temperature becomes 5 times, so temperature becomes 5T, so lambda will become 1/5, right? Because product must be constant, so lambda will become 1/5. Earlier it was 14, now it will become 14 by 5, so 2.8, very simple. So Wien's displacement law, very important. This one is also based on similar, but there is one more important formula here in this question that we'll study. So maximum wavelength is lambda naught. Now wavelength, black bodies now change, temperature is changed, so that maximum energy is emitted at 3 lambda by 4. So basically lambda t is constant, so lambda becomes 3 by 4. So what will temperature become? Temperature become 4 by 3, so that the summation is constant. So initially the temperature was T, now the temperature is 4T by 3, right? This is what we got from Wien's displacement law. Okay, but power radiated by the black body. So it is asking about the power radiated. So guys, what is the, now how do we find power? So there is one more law and it is called Stephen's law, which is about power radiation. Okay, and what is the formula? Power is equals to sigma T raised to the power 4. Very simple. So it says that it is based on T raised to the power 4. So if temperature becomes 4 by 3, the power will increase, of course, and it will become 4 by 3 power 4, right? If we find the ratio, so power, new power, it will become sigma 4T by 3, and power of this is 4. So sigma T power 4 and 4 by 3 power 4. So basically 4 की power 4 divided by 3 की power 4. So 4 की power 4 कितना होता है? 4 की power 4 होता है 256 divided by 3 की power 4, that is 81. So 256 by 81, that is the answer. So this is a very important law we have used here, Stephen's law, which is about power and temperature, very important. Okay. This one is interesting. It is a little bit counterintuitive also if you think about it. Sphere, cube and plate are made of same substance, okay, same mass, heated to 200 degrees Celsius, placed in a room. They will cool down, of course. Question is asking which will cool down at the last. Basically, we need to figure out which will cool quickly, which will cool slowly. So, like, how do we find that? Shouldn't they cool down at the same rate? Because, you know, they have same temperature, same mass. Why is the difference coming? Answer is, some of you might have incorrectly answered it as D, which is not correct. There is one change, one difference between the bodies which will cause a change in the timings, and that is, what is that? So the surface area are different, right? And more the surface area, more will be the heat conduction, right? That is, we also looked at this formula also. So if this is constant, so H by A, just focus on the area. So if we increase the area, the heat conduction will increase, right? If I send area to this side, so if area is more, the rate of flow of heat will be more. So that is the question about. It is about the rate of flow of heat because of different areas. So here, which will have the largest area, which will have the smallest area? So guys, we know that for a given volume, so the volume are same for all because they have same mass, same density, same volume. So for given volume, which has the minimum area, sphere. So sphere has the minimum area. So that is why it will conduct heat very slowly. So it will cool down at the last. So that is the answer. So interesting one, this one. Okay, constant power. Okay. This one, okay, let's go to this one, 18th one. Block of wood is, this one is also interesting. This is, which formula is, this is buoyancy law. Alright, so what it is saying, block of wood is floating in water at 0 degrees Celsius. Okay, temperature, we increase the temperature very slowly. What is the rise in temperature? What is the rise, with the rise in temperature, what about the volume of the block? So basically what it is saying, let's say there is a water body and there is a cube. Okay, it is floating. If I increase the temperature from 0, what will happen to this volume? V out. Okay guys, so this is interesting. Let's talk about this volume is, let's say, V in, which is inside water, and other volume is V out. Of course, V in plus V out summation is constant because volume of the body is not changing. We are heating, so temperature of water will increase. So guys, what is happening, why it is floating in the first place? Because of buoyancy force, right? So if I talk about buoyancy force, I can say that buoyancy force is equal to the weight of the body, mg. Okay, so what is buoyancy force? What is the formula? Formula is very simple. It is density of the liquid into volume of the body immersed. So that is V in. Okay, into the into G. So this is the also the weight of the liquid displaced, and that is mg. So G. So guys, what will happen if I increase the density for a second? If you think about it, if I increase the density, the volume which is inside, that will decrease, because the summation, the product is constant, right hand side is constant, right? Mass is not going to change for the block. So if I increase the density, the volume inside will decrease, and if volume inside decreases, it means it will come out more. So what I'm saying is, if we increase the density of the liquid, the block will come out more, and if we decrease the density, it will sink. So now let's look at the question. When we increase the temperature, what happens to the density? So guys, you might be knowing that the density of water is maximum at 4 degrees Celsius. So when we heat, the temperature will increase. So from 0 to 4, the temperature will increase, and density will increase till 4 degrees Celsius. And when the density is increasing, the volume inside is decreasing. It means volume outside is increasing. So till 4 degrees Celsius, the volume outside will increase, and after that, the density of water will decrease. So volume inside will increase and volume outside will decrease. So to summarize, from 0 to 4, the volume outside will increase, and after 4, between 4 to 10, the volume outside will decrease. So first increases, then decreases. That is the answer. Not simple. I would say this question, it required a lot of logical reasoning in terms of, but most important was this buoyancy formula. Without it, you cannot do anything, right? If you know this, maybe you can do the reasoning, you can talk about. And one more thing, which was anomalous expansion of water. The density is maximum at 4. That also you must know. Okay, guys. Next one. What is it is like almost same as this one. It involves Wien displacement law. It involves the Stephen's law, which is about power. Okay, so it is observed wavelength corresponding to maximum energy changes. So wavelength become half, it means temperature becomes twice. If temperature becomes twice, power is T power 4, 2 power 4. What is 2 power 4? So 2 power 4, yeah, so that will be, basically it will decrease, and yeah, so wavelength is the temperature increasing, so that is the power. So power will increase, that is why. So we'll basically to do the 2 power 4, that will be 64. So actually it should be 64. So power will become 64 times the initial value. That should be the answer. All right. Okay, let's continue. Okay, what is that? So this is again from calorimetry. It is saying tap supplies water at 15 degrees Celsius. Okay, that is how we mix water in winters, right? There is one tap which is supplying cold water, one hot, and we try to mix and maintain the temperature. Very difficult thing to do, but that is what we are trying to do here. So tap water supplies water at 15, which is the cold water. Geyser supplies at 95 hot water. How much water we should take? Hot water from the geyser we should take. So that total is 60 kg, and it is at 35 degrees Celsius. So guys, what we are saying is basically again calorimetry, heat loss equals to heat gained. Hot water losing heat, cold water gaining heat, both are same. So just apply the formula. So let's say hot water mH, okay, specific heat of water is going to remain same, into delta T. So what is delta T for hot water? Initially it was at 95, now it is at 35. So 95 minus 35, that is delta T. Again guys, positive values only, not the only the magnitude of delta T. Okay, that is equals to heat gained by cold water. So MC cold, okay, into specific heat of water into delta T. What is delta T for hot cold water? It is at 15. Finally, it is at 35. What is change? 35 minus 15. That is the change. So we'll do the calculation later. So this is 60. So specific heat gets cancelled. mH into 60 is equals to m cold into 15. This is 20. So 35 minus 15, 20 ratio is 3. So we can say that cold water, if we find the ratio, that is 3 times. So cold water must be 3 times. Okay, and hot water must be 1 by 3. Total is 60. So divide 60 into 4, 15, 15, 15, 15. So 15 kg, take the hot water, and thrice of that, 45 kg, that will be the cold water. So that the total is 60. That is the answer. Right? If you didn't understand it, you can also write one more equation, that is MC. Let me write it here. That is M cold plus M hot. The total is 60. So using these two equations, if you solve, you can get that M hot will be 15 kg. So that is the answer. Alright guys, so again basically calorimetry, this equation. Okay. This one is simple. Temperature increases. Okay, volume remains constant. So what happens guys, so when temperature increases, we know that the molecules will move more quickly, right? Their velocity will increase. So that is why higher velocity, that is the right answer. They strike the wall. Of course, they'll strike the wall more often, the amount of collision will increase. So that is the answer. Okay, so here what is happening? Volume of T. So this is guys now ideal gas equation. In such question, we have to apply ideal gas equation, which is talking about temperature, volume, pressure, all of that. So ideal gas equation is very helpful in such. Okay, air is filled 60 in a vessel of open mouth. Vessel is heated, temperature so that 1/4 of the air escape. Volume remains same, pressure also remains same because it is open. So pressure is same as the atmospheric pressure. So pressure, volume both are same, and both are same. We can say N1 R T1 is equals to N2 R T2. That is the formula we are going to use. N1 R T1. What is R and R will get cancelled. So okay, we don't need to write R. So N1 into T1. What is T1? So T1 is 60, but we need to convert it into Kelvin because that is the SI unit. So if I convert it into Kelvin, that is 273 plus 60 is equals to N2. So guys, what is N2? We know that 1/4 part of the air escapes. So 1/4, what was the air initially? N1 mole, 1/4 escape. So what is remaining? 3/4 is remaining. So 3/4 N1. That is basically N2 into T2. So here if we solve this, we'll get T2. What is T2? That will be 444 Kelvin. Is that my answer? No. Why? It is Kelvin, it is degree Celsius. Convert this into degree Celsius, you'll get the answer, which is 171. So one more important thing we needed to be careful in the last step, that was converting this to degree Celsius, because 444 is also there in the option. So yeah, question maker is trying to trick us, but we need to be careful with the units. Okay, this one. Pressure, temperature graph is given. It is asking about volume. So guys, remember this. So pressure, temperature. So okay, the graphs are given, the slopes are given. We can see this is at higher slope, this is at lower slope, right? The slope of this has more. So if we talk about slope is basically y by x. So p by t. So guys, what is p by t? So PV equals to nRT, right? We know PV equals to nRT. So what is P by T? P by T is nR by V. So slope is basically nR by V. N equal number of moles, N is same, R of course remains same, V. So we can say that it is inversely proportional to V. So what we can say from this graph is, if slope is more, V is less, volume is less. So which has more slope? 3 and 4. So 3 and 4 has more slope, so they have less volume. That is the answer. So 3 and 4, of course their volume is equal because they are on the same line. So we can say V3 equals to V4, V1 equals to V2, and V1, V2 will be more because they have less slope, so more volume. So V1, V2 is more than V3, V4. So that is why the answer will be this, that V1, V2 is more than V3, V4. So yeah, this is mostly about analyzing the graph and ideal gas equation, of course. Alright guys, next one, this is simple. Okay, what is work done? So guys, remember anti-clockwise. This is anti-clockwise. So in anti-clockwise graph, PV graph, the work done is negative. This is very important. In this negative work done, actually we are not doing any work, the gas is doing work on the system. So this is actually this is actually work done by gas. So gas is doing negative work. So gas is doing negative work in anti-clockwise. This is anti-clockwise, answer must be negative. What is the area covered? Is the magnitude, that is 20. If you calculate this area, so again unit, so 200 cc, what is cc? Centimeter cube. Convert into meter cube. This is kilopascal, which is 100 kilopascal. Okay, so which is 100 into 1000 pascal. So yeah, just multiply them. 100 into 1000 into centimeter cube. So centimeter cube into meter cube is 10 to the power minus 6. So that is the value. So this is pascal into meter cube. Pascal into meter cube is nothing but joule. So if you solve this, you'll get the answer in joule, and you have to put minus sign because it is anti-clockwise. And if you solve this one, you'll get minus 20. So this is important. And how did we find the work done? Area. Area gives the work done. PV graph, area gives the work done. PV graph. This is important. The PV graph, so that is why we can calculate the area. Not in this PT graph, of course. PV graph area gives the work done. Okay, next one. Okay, no work is done. Guys, this is first law of thermodynamics. What is that? Heat is equals to W plus delta U, right? No work is done, so Q equals to delta U. Okay, what is the formula of delta U? Change in internal energy. So guys, the formula is F by 2, that is the F is the degree of freedom, into nRT. So this chapter does have a lot of formula, but yeah, we need to go through all of them. We need to remember this one. Law of thermodynamics, very important. Change in internal energy, again very important. F by 2 nRT. Now just put, okay, monoatomic. That is why it is saying monoatomic, because it is giving the degree of freedom for monoatomic. What is that? 3. So 3 by 2 nRT. Put the values, get the answer. What is that? 2 mole. Temperature is also given. Everything is temperature, change in temperature. So 100. So put here delta T 100. You'll get the answer. Alright guys, 25 done, half of it is already done. Let's go to the other half. Okay, again guys, so this is positive work, this is negative work, because it is clockwise, right? And it is anti-clockwise. So which is more? The negative is more because it has more area. So negative work done is more, positive is less, overall it will be negative, that is why negative. Okay, now it is asking work done from C to A. So guys, we know that the work done is the area covered in the PV graph. But in this question, we need to be careful that V is on the y-axis. Generally, we take V on the x-axis, but here it is opposite, still fine. Basically, we'll measure this area. Area, we have to measure the area between the graph and the volume axis, which is y-axis here. So we'll measure this area, and that will be the answer. So what is that? So this is basically this rectangle plus this triangle. So if you look at the area, this is 1, this is 1, this is 9. Alright, so if you calculate the area of this, so 1 into 1 plus triangle half into base, which is 9, into height, which is 1, so 5.5. And as you can see guys, this basically whole thing is anti-clockwise. So we can say from C to A, the volume is decreasing, right? So if volume decreases, we know that the work done is negative, because gas is contracting, so work done by gas is negative, so that is why minus 5.5, that is the answer. Okay, next one. This is interesting. What we are saying is, if we blow air, what happens to the liquid? Okay, so basically guys, if we blow air, what happens? It is kind of like, you might have heard in the tornadoes. India doesn't see much tornadoes, but in US, in tornadoes, the rooftop, it disappears. How? So that is Bernoulli effect. The velocity is more, pressure will be less, because what is the equation? Pressure plus half rho v square plus rho gh, that is constant, right? H here is of course constant, so P plus half rho v square, that is constant. So if velocity is more, pressure will less. So when we are blowing air, we are increasing the velocity of air. And when velocity increases, pressure decreases. So air pressure will decrease and the liquid will rise more. Like that is how capillary work. If the above pressure is less, the liquid will go further up. So that is why level will rise above A, because we are decreasing the pressure by blowing air. So yeah, that is. Okay, next one, try this one. Okay, this is kind of a simple, I would say, if you know what is bulk modulus. So try this one, and we'll look at the solution in a second. Alright guys, so let's look at the solution. So basically guys, this is about bulk modulus. So guys, what is bulk modulus and what is the equation basically? So this is kind of like Young modulus equation. Stress is equals to some constant into strain. That is the general equation. So stress basically in terms of solid, stress is same as pressure force upon area. That is stress is equals to bulk modulus into strain. Here it is about volume, so delta V by V. Alright, so that is the answer here. So pressure is equals to bulk modulus. Okay, what is pressure? mgh, right, at the h height, rho gh, I'm sorry, when at the h height below a lake, the pressure is given by rho gh. Rho is the density of water, g height basically is the depth at which we are going. So pressure is rho gh. Okay, we'll put the values, that is k delta V by V. What is delta V by V? It is given, it is 0.1%. The volume is changing. Of course, it will the volume will decrease because of the pressure. So delta V by V will be 0.1%. So 0.1%. So basically divide by 100 to get in points. So rho gh, what is rho? Rho is 1000. What is g? G is 10 into h is equals to Young modulus. It is asking. Height is also given 100. So we'll put 100 height. And this is into 10 is power minus 3. Right? So yeah, just put the values, height is 100 and you'll get the answer of k, and that is this, option D. Alright, very straightforward I would say, just remember this equation, very important. K is the bulk modulus of course. Okay guys, next one, so 8 drops, it breaks into 8 drops, what is the work done? So guys, remember the work done, very important. Work done is tension into the surface area. Alright, that surface tension into the surface area. So if the surface area is changing, we have to do work. So here we can write delta A, the change in surface area. So surface tension T into the change in area. So that is how we can. So here the area is changing. Why the area is changing? Because the whole drop is dividing into droplets. So when the drop is basically getting divided, what we need to do is we need to conserve the mass of the water. And conserving the mass means conserving the volume. So initial volume, that is let's say 4 by 3 pi r cube, that is equals to, now 8 droplets are there, so volume of 8 droplets. So 4 by 3 pi r cube. Of course the radius will be different, the small r will be smaller because now there are 8 droplets. So if we do this, we can see that r is equals to 2r, right? 4 by 3 pi, 4 by 3 pi, it gets cancelled. We take cube root, so r equals to 2r. Using this we can find delta A. Initially what was the area? 4 pi r square. Finally what is the area? 8 droplets into the surface area of each, that is 4 pi small r square. Right, so small r we know it is r by 2, so this will be r square by 4. So now the final area will be 2 pi r square. So what is delta A? Delta A is 4 minus 2, 2 pi r square, and we put it here, so t into 2 pi r square. That will give us the answer. Alright. So yeah. Okay, next one. Okay. Two identical Y Young modulus are given in this ratio 3:1. They are loaded, so have same extension. What is the ratio of weight? So it's very simple. Basically, we can say that weight is proportional to, basically it is applying force. Okay, and force is basically, we are saying it is same. Okay, so force is same, which is mg, which is the weight, and we know that force is equals to Young modulus. Force upon area is equals to Young modulus into strain, delta L by L. Alright, that is what we are saying. So two identical wire are there, Young modulus are different. So it means length is same, area is also same, force is also same. So Y into delta L, that is going to be constant. Alright, that is going to be constant. So basically the, and here, so but okay, so here what is happening, guys? The weights are different. We are supplying different weight to different. The area is same because identical wire. Length is same. Extension is also same. It is given. Extension is also same. What is different? Force and Young modulus. So force I can write directly proportional to Young modulus. So if the Young modulus is in the ratio 3/1, the weight must also be ratio 3/1. So that is why 3/1. That is the answer. Here we can see F1 by F2, which is the weight, is equals to Y1 by Y2. From this equation we can see this, because A, L and delta L all are constant, same for both the wire, right? So this formula very important, formula of stress strain relationship. Right, guys, next one, change in pressure. This is again guys same formula, and that is pressure or change in pressure is bulk modulus into change in volume by total volume. So again formula this you can apply, right? It is asking change in pressure, but here the only difference is instead of change in volume they are talking about change in density. So we can find corresponding, not a big deal. If we know change in density, we can find the change in volume. Alright, so ratio will be same actually. Delta V by V will also be 0.1%. Delta V by V 0.1%. Young modulus we know, we can find the pressure very easily. Alright guys, next one. This is very simple. We know the potential energy is equals to half k x square. So we can say it is proportional to x square. X is the elongation. So x becomes what? 5 times. So u will become 5 square, 25 times. So half u equals to half k x square. Spring potential energy, that is the formula, very simple. Okay, next one. Okay, again that formula which was there, Young modulus formula, we have to apply. That is force upon area is equals to Young modulus delta A L by L. So guys, again this is very similar to this question, this one that we did. Okay, so if you understood this one, I think you can also do this one. Forces are same. Here what is different? Area is different and length is different, and we need to find the elongation ratio. Young modulus is same wire, same material. So yeah, area is different. How do we find area? So here guys, area A1 L1 is equals to A2 L2, because A1 L1 is the volume of 1, A2 L2 is the volume of 2. Volume are also same. It is given same mass, so it means same volume. So from here we can find ratio between A and L. Using these two formula, we can find the answer. Alright. Okay, let's go to the next one. Speed is given. Okay, and 3600 wave pass over certain point in 1 minute. So in 60 seconds, how many waves are passing? 3600. So in 1 second, how many waves will pass? 60, 3600, 3600 by 60, so 60 waves. So in 1 second, 60 waves are passing. In 1 second, n number of waves is passing. So what is that? That is frequency, guys. So frequency is 60. 60 hertz frequency. Speed is given. You can find wavelength, right? I hope you know the formula. Speed is equals to wavelength into the frequency. Very important. So everything is given. We need to find wavelength, frequency we have found out. So yeah. Okay. Okay, this weird equation is given and they're asking speed of the wave. So guys, this equation is the equation of wave, and this is, we need to know the general form, and that is omega t minus kx. Minus or plus, both are fine. And this is just the phase, we can ignore the phase. So omega, what is omega? Omega is 2 pi by 2 pi into frequency. Right, 2 pi into frequency, that is omega, and that is 7 pi. Right, you can compare. Omega is 7 pi. So omega is what? 2 pi into frequency, that is 7 pi. So frequency is 7 by 2. That is first thing. Now this is k. K is 0.04 pi. So we'll compare. So what is k? K is nothing but 2 pi by lambda, and that is 0.04 pi. So we'll compare, and now we can write lambda, which will be pi and pi get cancelled. This is 2 upon 0.04. So that is 50. So wavelength is also there, frequency is also there. We'll multiply and we'll get the speed, because speed is nothing but frequency into wavelength. So yeah. Next one, phase difference is asking guys. So one equation is this, a sine omega t plus pi by 6 is what? 30 degree. What is the other equation? A cos. So it is cos. We cannot compare directly. Can I write it in sine form? Yes guys, cos and sine can be easily converted. So I can write this thing like this. This is omega t plus 90 degree. Because what is sine 90 plus theta? Sine 90 plus theta is cos theta. So this cos omega t I can write like this. A sine omega t plus 90. Now we can compare. This is omega t plus 30. This is omega t plus 90, what is the difference? 60 degree. So that is my answer, 60 degree, which is pi by 3, right? Pi is 180, pi by 3 is 60. Okay. Now it is about amplitude, ratio of amplitude. So we need to remember, guys, basically when the points are away, so this is a point source, so the intensity will expand, and intensity will be inversely proportional to the distance of the square, right? So if the point is 2 meter away, the intensity will become 1/4, and we also know that intensity is proportional to amplitude square, right? So if we compare the two, we can write that amplitude square is inversely proportional to square of the distance, and thus if we take under root, so amplitude is inversely proportional of the distance. So if the distance is double, amplitude will be half. So that is the answer to this one. Distance is 1:2, 1 meter 2 meter, so amplitude will be opposite of that, that is 2:1. So that is the answer. Wave is going, so this I would say the most easiest question of the test. When we know when a wave goes from one medium to another, frequency doesn't change. This is factual. We need to remember this one. Frequency doesn't change, wavelength changes, velocity changes. Alright guys, next one. So this is about speed. So yeah, frequency is given and wavelength is given. So we can find the speed and what is the speed of the wave, frequency into wavelength. So that is 25 meter per second. So now it is asking about time. Let's look at it. Time taken to find cover distance 8 meter. So time we know is distance upon speed. So distance is 8 divided by speed, 8 by 25. And what is that? It is going to be, if you calculate, around 0.32 second. So it means statement 1 is correct. 0.32 second. This is correct. Okay. What about statement 2? Time taken by point on the string. So this is the speed of the wave, right? Wavelength into frequency. What about a point on the string? So like if a wave is there on the string, we know that this point in the string, it is doing SHM. It is going this, like this. So is the velocity of this point is also same? No. Is the wave velocity is not same as point velocity. So the velocity or the speed of this point is not 25. It is different. So that is why time it will take to cover 8 meter is not 0.32. It will be different. Whatever doesn't matter because the speed is different. So yeah, we can directly say that statement 2 is incorrect. So that is the answer, option B. Alright. So yeah, just remember that wave velocity and speed particle velocity are different things. They are not the same. Okay, transfer wave is there. Equation of wave is equal to shape of the string. So equation, what is equation, guys? Equation is sine function omega t. Sometimes it can be sine, let's say. So what is the wave? Wave will be like that. It is the sine graph. So the equation tells me what is the basically the shape of the string. So this is correct. Equation gives the general displacement of each particle. So this is correct. Must be sinusoidal. Not necessarily. It can be other function also. It can be cos function also. Not necessarily. Equation is displacement at one end only. So that is not correct. It is equation displacement at each point on the string. Not just one end, but all the points in the string. This equation can tell me what is the displacement at all the points. So this equation is not about one point, but all the points. So that is the answer. Okay. This one is what is the topic? So Doppler effect. Very interesting phenomena. So guys, we need to remember here what is happening. Man is watching two trains. Okay, one is coming like this, one is coming, one leaving. So one is going like this, one is coming like this. Let's say man is here. So what will be the frequency he will listen? So frequency is the original frequency into velocity of sound divided by. So this is, let's say, first source. So source is coming. So due to this, the frequency will increase. So denominator should decrease. So we need to subtract the velocity of the source. That is the first due to F1. Okay, let's call it F1. What about F2? F2, guys, the frequency will be less because the source is going away. So it is frequency into velocity of sound. This is velocity of sound. Okay, V plus Vs. Vs is the velocity of source. V is the velocity of sound. So these are the difference in frequencies. So we can find the value easily. F is 240. V is speed of sound 320. Vs is 4. Okay, and F1 minus F2. That is my answer because that is the number of beats that per second. the difference in the frequency. So I can find it using these two and putting the values I can find very easily, and this will involve some calculation that we can do. If you solve this one, answer you will get option A. Right? I'm not putting the values, that is not the difficult part. Difficult part was writing the formula and realizing that this is Doppler effect, so we need to find F1 and F2 different frequencies. And beats, important formula, the difference in frequency will give me the frequency of the beats, number of beats per second. Right guys, next one, car emitting sound frequency 500, fixed wall. So again it is kind of similar. Observer is here on the car, so car is emitting sound, so car will go to the wall and it will come back. So the observer is hearing two frequencies, one is 500 Hertz and other is which is coming from reflection, so that will be different. So according to this observer, the source is moving, so what will be the frequency? V minus Vs. The source is moving, but in the second case, when the reflection is coming, then he is moving towards the reflection. So this is the velocity reflected, this is the frequency of reflected wave, and after reflection, the frequency that he will receive is, we have to multiply the velocity of the observer, that is V plus VO. Vs, VO both are same because source and observer, the car and the boy both are together, both are moving with same velocity. So that is the frequency that the boy will listen after reflection. Other frequency is F, the difference between them, that is the number of beats, and number of beats is what, 10. So we can subtract F minus this, and that is equals to 10, and we'll put all the values. F is given 500, V velocity of sound we need to find, Vs is what, 4, VO is also 4, put the values, get the answer. Simple, not that simple, but still doable calculation. Alright guys, next one, okay. This is simple, directly Doppler effect we can say. Source move away. So when the source move away, what is the frequency? V upon V plus V source. That is the frequency. This is 6 by 7 times of the original frequency. So we can see that V by V plus Vs is equals to 6 by 7. So here we can find Vs from here. So if you do the calculation, so Vs will be V by 6. So V by 6, what is V? 330 divided by 6, that will be your answer, B. Okay, next one, this intensity is given and they are asking the volume in decibels. So guys, this is very important formula of loudness of sound. So guys, what is loudness of sound? That is 10, okay, log, it is logarithmic function, bracket I, intensity of sound, divided by I naught. I naught is a constant, it is a standard reference level. This is the reference level. So I naught is this, 10 to the power minus 12 given in the question. Even if it is not given, we can remember it. It is 10 to the power minus 12 watt per meter square. I is given 10 to the power minus 6 and 10 if we put the log function. So this is 10 log, inside we have 10 to the power 6. So power will come outside, it will become 60. So 10, base is 10, so that is why this will become 6 into 10, 60 decibel. That is my answer. Alright. Option C. Okay, which of the following does not represent? So guys, this is a 10 function. 10 function is very weird kind of a function. If we draw the graph goes like this, right? At 90 it becomes infinite, just after 90 it becomes this. So this is a graph of a 10 function. It is not sinusoidal. This is fine because it is combining two waves. It is kind of like interference. This is fine, this is fine, this is not fine. 10 is not a SHM function. Alright. Displacement is given, value of time period. So guys, we can just compare. So A sine omega t. So omega is 20 and we know what is omega. Omega is 2 pi by t, that is 20. So here we can find t, that is 2 pi by 20. So pi by 10, that is my answer. Very simple. Comparing, remembering the general equation, A sine omega t plus phase, which is 60. Phase doesn't matter with respect to time. Okay. Average velocity of particle in one time period. So average velocity, guys, what is average velocity? Total displacement upon total time. In one time period, what is the total displacement? So if you look at one SHM, particle goes like this and then comes back. So it comes back to the same point in one SHM. So total displacement in one cycle is 0. That is why average velocity is 0. Okay. So this is very similar to we did earlier, u proportional to x square. X becomes 3 times, u becomes 9 times. answer. Straight 10-second question, right? This question exam should take you like 10 seconds. Time period of a single spring pendulum is T taken on moon. What is the time period? The trick question, what is the time period of spring? This is not a string pendulum. This is not a simple pendulum. This is spring pendulum. And know that when in the SHM of a spring, what is the time period? That is 2 pi root m by k. K is the spring constant, m is the mass which is in SHM. So nothing to do with acceleration due to gravity. This is not a simple pendulum. This is the spring pendulum. This is the time period we did. We have studied this time period of spring. SHM is this. So moon, earth, Mars doesn't matter. Time period will be same everywhere. That is why answer is equal to T. Alright guys, that is it. We have covered all the physics question, and earlier we have covered all the botany, zoology and chemistry questions. Alright, so that was minor test 2, which was conducted a day before yesterday. After this we have minor test 3, which will include the syllabus of 12th class. What we are trying to do is we are trying to help you cover all the syllabus of the NEET so that you can get a really good rank in the NEET. That is why we call this series All India Rankers Test Series AIRTS. So if you want to get a good rank, please do the papers diligently, follow the discussion here, and that will be really helpful for the NEET exam that is just about to come. Alright guys, that is it from our side today, and thank you very much for joining. Thank you very much for your attention, and I wish you a good day ahead.